Hello, my name is Alexander Mardvitsa. I work at Google Research, where I lead a little team that works on self-organizing systems design. Today, I would like to present our new work on isotropic neural cell automata. Our work is biologically inspired, and we are looking for practical ways of designing systems composed of multiple locally interacting agents that are capable of solving complex global problems. One of the most important goals that we are trying to achieve is to develop a realistic computational model of a multicellular organism development. Most of our work is based on applying the principles of differentiable programming. We represent agents with tiny differentiable neural networks. Then we put those networks into a simulated environment and allow them to interact for some time. Then we apply a designed objective function that evaluates the behavior of the population of agents and measures how closely does it match the target behavior that we want the population to learn. And then we use backpropagation to optimize the agent parameters to steer the population into the following the desired behavior. Isotropic neural cell automata extend our work on growing neural cell automata and follow-up work on self-organizing textures. The original growing neural CA model had a problem that I consider to be very annoying. If we take a developed pattern and rotate it by resembling states of the grid, it explodes, trying to rebuild the original orientation of the target lizard. Directional preference is called anisotropy. It's known that many classical cell automata models, such as Game of Life, are capable of handling 90-degree rotations or reflections without disturbing the functional pattern, so they are partially isotropic. This anisotropy is caused by Sobel filters that are used as a part of cell's perception. I prefer to think about those filters as pairs of sensors that measure change in cell's state in the neighborhood of each cell along a particular direction. Those filters are aligned for all cells on the grid. We can actually synchronously rotate those sensors to rotate the pattern or even deform it. One of the ways to make neural CA anisotropic is to avoid using Sobel filters, which essentially give cells sensitivity to the absolute direction and magnitude of the gradient in the neighborhood of the current cell. So the simplest way to make neural CA anisotropic is to remove Sobel filters and keep only using Laplacian kernels, which, by the way, is easy to generalize to various grid structures or even non-regular grids. To make cell perception richer, we can also give it access to Euclidean norms of the state gradient vectors, which are rotation invariant. We can also remove neighborhood perception completely and give cells access to their own states only, but also add a separate diffusion process that works in parallel with cell updates. This is essentially a variant of a popular reaction diffusion model, which is known for its capability to generate diverse interesting patterns. The famous linear model can also be seen as a case of isotropic cell automata that relies on convolution with large symmetric kernel to model cells' perception. Now let's get to practice. First, I'll try to modify the texture synthesized neural cell automata to make it isotropic. But let's first look at the behavior of the original anisotropic CA. The target texture pattern is fairly isotropic, but we see that there is some direction of preference in the generated video. For example, spots of different colors drift in specific directions. The first attempt to make say isotropic differs only in a set of filters used for perception, and also I changed the sizes of CA parameter matrices to accommodate a change of input size and also give it a little bit more parameters to compensate for missing filters. And here is a video of training dynamics. The model was able to reproduce the characteristics of this pattern, but the quality of color reproduction is worse. Let's enhance the cell perception by giving it access to the Euclidean norm of the gradient vectors. This model trains faster, it produces textures of better fidelity. This method works well for textures that don't have particularly strong directional preference and more or less isotropic. But what if we try another example? A texture which has strong directional characteristics. It's not surprising that the optimization couldn't find a CA that reconstructs the pattern properly because cells don't know where left, right, up, and down is, but the objective function is asking for a texture pattern having a specific orientation. In our differentiable programming of reaction diffusion patterns article that we published at ALife conference last year, we proposed a simple method to fix this problem. We match the current pattern against pre-computed, reflected, and rotated versions of the target, and then select the orientation that produces minimal loss for backpropagation. This way we managed to train isotropic reaction diffusion models, which are also case of isotropic neural cell automata that are capable of reconstructing anisotropic textures. Such isotropic models have a number of attractive properties. For example, they can be easily applied to meshes or other manifolds that are different from the training regime. They can even be lifted to a space of higher dimensionality without retraining. All they need is Laplacian defined in this space. We face a similar problem when we plug in isotropic NCA into growing narrow CA setting. So we want a neural cell automata that builds a target pattern starting from a single seed cell. But due to the lack of directional sensitivity, all it can do is to grow something that's average across all rotations and reflections of the target pattern to minimize the loss function. Eventually, it learns to exploit the raster nature of the grid to have this fourfold symmetrical pattern as a result. But we want isotropic system to grow the highly anisotropic target. So how do we do that? The first strategy that we propose in the growing isotropic neural cell automata paper that we submitted to a live conference this year is based on using structured seeds. We use at least three differently colored points as a starting configuration for the pattern development. And the relative orientation of these points pinpoints the orientation and reflection of the target pattern. So CA can build it in a specific orientation. After training, we can vary the orientation of the resulting pattern by varying the placement of seeds on a grid. We can even produce variations of the target pattern by using out of training combinations of starting seeds. Sometimes results can be a little bit unexpected. Another training strategy that can train models that can build patterns starting from just a single seed is based on rotation, reflection, and variant loss. This loss transforms both target and neural C output into polar coordinate systems and then uses matching in Fourier space to efficiently match patterns against all rotations that become horizontal shifts in polar coordinate space. And then, similar to the strategy that we were using for textures, we select the minimal loss for backpropagation. And this procedure is repeated for the original pattern and for reflected pattern. This method allowed to train isotropic CA that was growing highly anisotropic pattern by breaking the symmetry through amplifying the noise coming from randomized cell updates. So each time it was growing a differently oriented lizard, but some symmetries happened to be harder to break than others. For example, often our training was resulting in a model that had this ghost image of upward and downward looking lizard. We managed to fix this problem by augmenting the target pattern with extra channel that has strong asymmetry. And we can see that self-similarity plot for the original pattern contains this strong local minima corresponding to 180 degrees rotation. And the augmented pattern has a much smaller local minima corresponding to this rotation angle. This video shows the dynamics of a single seed isotropic model trained to grow anisotropic pattern with rotation, reflection, and variant loss. We see that in spite of identical initial conditions, we get random orientations of lizards, and even chirality is not determined at the model, so we get mirror reflections. And this video shows one of the early training checkpoints that displays some artifacts and instabilities that I found particularly entertaining. In addition to our earlier experiments in generalizing models from square to hexagonal grids, we tried to generalize this model on a non-regular grid. So we use Poisson disk sampling to generate the distribution of cells on the grid. Then we compute Voronoi diagram, and our Laplacian operator computes the difference between cell state and the average weighted average of states of its neighbors, and weights are proportional to length of shared edge between two cells. This video shows an example of run of this model trained on square grid adopted to a non-regular grid. The first run manages to produce coherent pattern by breaking the symmetry of the growing front. The second run produces a two-headed lizard first, but later recovers to a normal configuration through a complete remodeling of the pattern. Both last year's reaction diffusion and this year's growing isotropic neural CA papers are available online, and the code for reproduction of isotropic and CA experiments will be published a bit later this year. Thank you for your attention, and I'll be happy to answer your questions.
That's fantastic. Thanks, Alexander. Okay, so let's open the floor up for questions. If you want to raise your hand, I'll get to you in turn. So use the reactions at the bottom of the Zoom. Any questions? No questions. Seth's asking, have you considered extending the model to 3D? I'm not getting your audio, Alexander. I wonder if Sylvia, have you changed something with the audio settings? I can't hear Alexander at all.
No, but I can activate its own. Or Alexander, are you active now? It's active.
Can you hear me? Can you hear me?
Yes, now we can hear you.
Okay.
Good.
Okay, great. Well, we didn't yet do the full 3D training for neural cell automata, and the reason for that is actually we're in the process of transition from grid-based models to particle-based models, because I think in higher dimensions it's going to be more resource efficient, and also, well, I personally find the particle-based models more appealing. So I hope for 3D we'll be able to move away from raster grids. But yeah, that's definitely on our roadmap.
Francesco is asking, is it always needed to have more than one initial cell to break the symmetry?
Well, we demonstrated that actually we can break, we can use stochastic updates to break symmetries. So one cell is sufficient, as we've shown in our last experiment. Actually, we also, during making this paper, we tried to demonstrate that symmetry can't be broken if all cell updates are fully synchronous. And surprisingly, our models were capable of breaking the symmetry with fully synchronous updates from a single cell. And the reason of investigation that we discovered, it was happened to utilize computational loss of precision in the order Laplacian filter accumulates the neighborhood of each pixel. So basically, eventually we managed to implement Laplacian filter in a fully symmetric way up to numerical precision, and this way the system finally couldn't break the symmetry. Well, noise is sufficient source of randomness to break the symmetry. But in order to well, in structured seed experiments, we had to use 3 seeds because our models perception was completely symmetrical. And actually, we had a variance of models that I didn't include into the paper presentation, which I'll talk about it maybe in the next article, which are called steerable models that have chirality, where each cell has an own direction vector that it controls, and those were capable of building target pattern seeded by just 2 seeds, and chirality was intrinsic property of cells perception.
Okay, thanks, Alexander. So actually, we're just out of time for questions. So Lana has asked a question in the chat. So when the next talk's running, maybe you could reply to her either directly or to everyone in the chat. Thanks very much for your presentation, and let's everyone give him a round of applause using the clap. How about that? There we go. Thanks, Alexander. So the next presentation is by Christopher Bennett, Seth Bullock, and Jonathan Laurie, and it's on exploiting intrinsic multi-agent heterogeneity for spatial interference reduction in an idealized foraging task. And it's really hard to say in one breath. Go ahead, Seth.
Thanks, Alan. Hello, everybody. This is work by Chris Bennett, who is a PhD student supervised by Jonathan Laurie and myself. And the topic is heterogeneity. So when we think about multi-agent systems, robots or software agents, we're often thinking about swarms, large numbers of agents, and all of the agents are essentially interchangeable. But when you talk to engineers or companies that are building autonomous systems, the picture is often somewhat different. So they often want to talk about platforms plus people, so robots that interact with people or software agents that need to interact with people. And they also are interested in building products that form part of extended systems. So they're not individual products that stand alone. They are systems that have to interact with pre-existing systems. So they're often thinking about heterogeneous situations and heterogeneity. And so that's changing our thinking from swarms to squads. So maybe a set of agents that have to work together, despite those agents being fundamentally different from each other in important ways. And we know a little bit about heterogeneity, but it's actually something that is surprisingly underexplored within multi-agent systems. And ALife is a good setting, I think, for thinking about heterogeneity. We know a little bit about it in social contexts, so we know it can be positive. For example, the wisdom of the crowd. Many individuals may be quite poor at answering a question, say how many sweets are in this big jar, but collectively they actually do a good job of that, despite the variability within the population. And on the other side, we know that heterogeneity can be damaging, and we spend a lot of effort to ensure uniformity and regularity in engineered components, for example. And in biology, the same picture. We know that heterogeneity can be very powerful and positive. So in these social spiders, the fact that the spiders have varying personality in terms of how brave they are improves their collective exploration and foraging performance. But on the other side, we know that heterogeneity is something that biology sometimes fights against. So your T cells are looking for cells in your body that are too different, and they're killing them in order to cope with, for example, something like cancer. In the robot setting, the sources of heterogeneity that we might care about are, the example that I gave of the X-Men, they're all functionally very different, and that's one source of heterogeneity. You might have a set of robots or a set of software agents that are fundamentally different from each other. One robot has infrared sensors, one robot can fly, another robot has legs, but there are more subtle sources of heterogeneity as well. So you might have a set of robots that are all apparently the same, but they arise from, they have a different vintage. So your robot from 2008 might have different firmware, slightly different specification. The sensors that it uses might be slightly different from the robots that you've got more recently. And even if the robots originally were exactly the same, long-lasting changes like failures or faults can introduce heterogeneity to a system. And if we go back to the idea that robots might need to interact with people, a hybrid system like that is a combination of people and robots, is always going to be fundamentally heterogeneous because the people are not the same as the robots. And we've already stumbled into a distinction that is maybe not made enough in the literature between what we're calling intrinsic heterogeneity. So that's agents that differ in something that is relatively permanent, something that's inherent to them. For example, one person is colorblind and another person is not. That's an intrinsic property, compared to extrinsic heterogeneity. So more fluid, more plastic changes. So one robot might be assigned task 1 and a different robot might be assigned task 2, but later on those task assignments might change. So that heterogeneity is not fixed, but is more plastic. And that second class of heterogeneity is much more studied. It's not often talked about in terms of heterogeneity, but it's much more familiar in multi-agent systems. Intrinsic heterogeneity is a little bit more rare. So ultimately, we're interested in trying to design to exploit heterogeneity. If it's going to be there, if it's almost a fact of life for these robot engineers, then we have two options. One is to extinguish it and limit it as much as possible, and the other is to somehow make use of it and exploit it. So when can we do that, and how can we do that? So we're going to look at that question in the context of a very simple model. It's inspired by a paper by Richard Vaughan and Casper Stoy and colleagues, and it involves a very simple spatial task. So the robots in our model shuttle backwards and forwards between home and food. So they constantly are just moving backwards and forwards ballistically. And when they're in a kind of free space area in the middle here, they can pass each other freely. They don't even notice each other. But in these restrictive tunnels, they can't pass each other. And if two robots meet going in opposite directions, one of them has to back up to let the other one pass. And if two robots meet going in the same direction, then if the faster robot is behind the slower robot, the faster robot can only move at the speed of the slower robot. And the overall collective performance that we're interested in is how many journeys can happen in the period that we simulate. So that's like how much food was collected during each simulation. And in the work by Richard and Casper, they provided this quite nice graph. what we expect is, as we increase the number of agents in this kind of system, the performance might initially go up because you have more agents doing more work, but the spatial interference caused by the tunnels is gradually going to limit the performance until eventually the interference starts to prevent any work from happening. And in their paper, when they were considering homogeneous agents, all the agents were fundamentally the same. And one of their results was that establishing a kind of fixed dominance hierarchy just at the beginning of a run, saying, well, you have the highest priority and you have slightly lower priority, was effectively as effective as tossing a coin whenever robots encountered each other. So what we're interested in is a similar model where we build in intrinsic behavior and try to explore not whether we can optimize performance for this task, but whether we get some insight into how heterogeneity can be exploited. So our agents have three behaviors. When they are in free space, so they're not in a tunnel, or when there's no other agents near them, they can navigate. So they're green and they just navigate forward at their maximum speed in the direction that they want to travel. But if they meet another agent in a tunnel, if they both collide head on, then they both become afraid, which is this yellow state, and they calculate their priority somehow. And if an agent can't see another agent that is higher priority than it, then it becomes brave. It becomes this red state and it moves forward. And if it doesn't become brave, it remains afraid and it will retreat to let the brave agent move forward. And whilst it's retreating, so there's an issue that can happen in this case because they have a limited visual range. You can have a situation where two robots become brave because they can't see the other robot that's involved in a kind of multi-agent collision. So in this example, for example, maybe 4 can't see 5. 5 is too far away. So 4 thinks I have higher priority than 1 and 2. So I should be brave. 1 and 2 should back off. 5 can maybe see 3 and 2 and 1. 5 could see all of these robots and believe that it is the highest priority robot, and that 3 and 2 and 1 should back up. So it's a bit of a sticky situation for robot 2, for example, who just can't win in this situation. So the mechanism here is that during this kind of deadlock, the robots gradually increase their maximum visual range up until its maximum. So whilst they're navigating, whilst they're happy, they have their visual range set to a minimum. They don't really care what's around them. But when they get into a deadlock situation, they gradually look further away and recalculate whether they're brave or afraid. And that should resolve most deadlocks. So to resolve conflicts, we have invoked this concept of priority. How is priority going to be calculated? Well, we have 2 kind of null models for that, 2 random assignments. Transient random means every time a collision occurs, we randomly and arbitrarily assign priority to the robots that are involved. So that gets done many times during a simulation and is essentially tossing a coin. But everyone gets a unique priority rank in a collision. And the other random scheme is that at the start of the simulation, we assign a fixed priority to each agent, and that holds for the entire simulation. So those are our 2 sort of null models. And then we have models where priority is assigned based on an intrinsic agent property. So an obvious way of doing that, given that our collective performance is how many journeys can happen, you might say, let's prioritize the fastest agent. So what happens is, in a collision, the fastest agent will have highest priority. We can also do the opposite. We could prioritize the slowest agent, and they could have highest priority when they encounter a collision. And we also have this second property, which is the visual range that a robot has. So in a system where the maximum visual range of a robot is different between the robots, when there's heterogeneity in the maximum visual range, we could prioritize the agents that have the maximum, the most far-sighted individuals. Or the opposite, we could prioritize the most myopic individuals that have the lowest maximum visual range. So remember that the robots are not always using their maximum visual range. When they're happy and green, they're just using their minimum visual range, and they only ever increase their visual range during a deadlock. So you can see that there are 2 relevant intrinsic properties that might be heterogeneous, maximum speed and maximum visual range. So here's what the system sort of looks like. Here we're prioritizing the fastest agents. So the number is their priority. Agent 7 is the fastest, has highest priority. Whenever agent 7 collides with a robot that is going in the opposite direction, it kind of beats it, and that robot has to retreat. By contrast, agent 1 is the slowest, has lowest priority, and whenever agent 1 collides with another robot, it ends up having to back up. So it's kind of a tough life for agent 1. He doesn't get to go forward an awful lot. So here's some of our results. First of all, let's look at prioritizing agents by speed, letting the fastest agents go first. So here we can see what happens in many runs with 2 agents, 3 agents, 4 agents, etc., all the way up to 9 agents. And what we're plotting here is the amount of food or the number of journeys performed by each agent. And you can see as the number of agents goes up, the number of journeys performed by each agent goes down because of the spatial interference. And you can also see that there is a spread of the number of journeys performed by agents within each scenario. And in fact, the largest number of journeys are performed by the fastest agents, and that's like a perfect correlation. We can compare that to our slowest goes first scenario, and we see the same sort of decrease caused by the spatial interference. Interestingly, the medians are not too different from the medians in the first graph, the kind of red horizontal bar. And the other apparent property of this graph is that that range of performance has completely collapsed. Basically, every agent now gets to do the same number of journeys as the slowest agent. And when we compare the overall performance of those two schemes, there's no statistical significant difference between the overall performance. So perhaps surprisingly, slowest first does just as good as fastest first when you consider it over a range of different population sizes. And when we compare that to our null models, they both do better than the coin flipping transient random scheme. But the best scheme is to establish a fixed random dominance hierarchy at the start and just employ that throughout the whole simulation. So surprisingly, perhaps, fastest first in a scenario where moving quickly is important turns out not to be the property that should be used to break deadlocks. Now you can see in these graphs that the distribution of data is not very different. So although these are significant differences that I'm pointing out, the size of those differences is not huge. And so far, what we've been looking at in every scenario, the average speed of the population is 50, and the difference in speed between 2 agents that have the closest speed is 5. So if there were 3 agents, we would have 45, 50, and 55 would be the speeds of those 3 agents. And we can increase the heterogeneity in the population by increasing that delta V to 10. So now we have a wider range of speeds, performance drops, and now fastest first does become the best scheme. And the overall performance using that scheme is better than slowest first, and both of those are better than the null model. So far, the visual range, the maximum visual range of all those agents has been identical, but now we can have speed varying and maximum visual range varying. Speed is now assigned at random, and maximum visual range is assigned at random, so they are not correlated. And in this circumstance, predictably, the agents that can see furthest should be prioritized in these deadlocks or in any kind of collision. But more interestingly, we can look at a situation where the speed property is correlated with the maximum visual range. So now we can have scenarios like this one where the slowest agents are also the most myopic. And if we let those go first, that is actually the worst case. And down in the bottom right, we can have a situation where the fastest agents are actually the most far-sighted also. And that is the best, kind of predictably, that's the best way of breaking these collisions. But the most interesting result is that the situations where those two properties are negatively correlated, so down the bottom left, the slowest agents are the most far-sighted, and prioritizing those is significantly better than prioritizing the fastest agents who are the most myopic. So that gives us an insight into the fact that the visual range is the dominant property. So in a scheme where speed, getting things done quickly, might have inclined an engineer to pick speed as the property that you would use to establish priority, actually the maximum visual range, because that determines how collisions are dealt with, and spatial interference is the dominating property that limits performance, it turns out that that source of heterogeneity is more important. So I'm just going to finish up very quickly. The results show that heterogeneity can influence collective behavior positively, but exploiting the heterogeneity in order to achieve that isn't straightforward. The extent of the heterogeneity matters, the types of the heterogeneity matter, the interactions between the heterogeneities matter, and they appear to be very domain specific. So if you care about a different task, not this kind of contrived tunnel foraging task, then you can't learn very much from the schemes that we're using. Designing for heterogeneity remains a useful goal, but the design principles that might be broader than one particular setting are elusive. Things to do next. Ecology actually has a very more advanced notion of heterogeneity than we've used so far, dividing it into different kinds of part, and we're interested in exploring those. We've so far just looked at intrinsic heterogeneity, but they clearly, intrinsic and extrinsic clearly interact, and we should be considering them together. But the most interesting idea that Chris has had, I think, is that given this domain specificity of the impact of heterogeneity, maybe we could employ something more generic, like a squad level empowerment measure. If you saw Christoph Salge and colleagues talk on the first day, empowerment is a way of measuring how influential an agent is on its environment. Could we bypass a deep understanding of heterogeneity in a problem space and use a more generic agent kind of information theoretic measure to drive this kind of prioritization scheme? So those are the questions that we're going to explore next. Thank you for listening, and thank you to my funders, and thanks to Chris for doing all the work.
Thanks very much for the talk, Seth. I saw Steen had his hand up really quickly there, so we've got time probably for 1 and a bit questions. Steen, go ahead.
Thank you. Yeah, really fun study. So I'm sure you're aware of traffic. There's been a lot of studies on traffic. So I was wondering. I have a question. So are you in after the phase transition? Because if you have very few agents on the road, then as free flow. But at some point, you start to see jams. So I suppose you are in the jam area, and we know from traffic that platoons is the solution. So instead of mixing, all kind of trucks and Formula One race cars together, you need to have these guys to communicate, so they form platoon. So you have a long sausage going through one way, and then another sausage going through the other way. Then we know you can increase the flow dramatically. So it's actually communication or cooperation rather than diversity or that would increase your flow. That would be my guess.
Yeah, so I think if we were interested in optimally solving the problem, you're absolutely right. And in some sense, the second graph that I showed you, the slowest first graph, where the performance of each agent was very similar, they effectively are platooning in that situation. The slowest agent ends up blocking all the agents behind it, and they all move forward at the speed of the slowest agent. In the other scenarios, because they're not sophisticated enough to slow down, as soon as they have the opportunity, they move as fast as they can. And they in the free space in between the tunnels, they ruin any platooning that was established serendipitously. But you're right, a more sophisticated agent would rely on some kind of communication and coordination to solve this task.
Yep.
Okay.
In fact, maybe, well, yes, great, sorry.
No, I think we need to move on just to get to the time. Thanks again for the talk, Seth. So I noticed that I think it was James Stovold had his hand up before. So James, if you could type your question in to Seth, maybe directly or to everyone in the chat, I'm sure he'll be able to answer it. Okay, so our next talk is from Arend Heinz, who's here online for a paper with Jory Schossau towards an FPGA accelerator for Markov brains. Off you go, Arend.
Yes. Thank you. You can see my screen, right? Yeah.
Yeah.
Okay. Perfect. So yeah, thank you. I'm going to talk about an FPGA accelerator for Markov brains. One of the referees pointed out at some place that, yeah, you're not having a lot of results, but the story is interesting. And I have to absolutely agree. So the subtitle here is There and Back Again. That's Bilbo Baggins' title and from Lord of the Rings. It's really about the journey that we went through here. So let's start. We of course all know artificial neural networks, and maybe you love them or maybe you hate them, but it doesn't matter. It's pretty much the predominant tool in AI applications in the AI research. And of course, artificial networks are probably so famous because they're really accessible, meaning you can run them on all computers that you have. And at the same time, they're also really fast because most of those computers have GPU accelerators, and it's a really cool technology to do this. So let's take a look at why this acceleration actually works here. This is the middle layer of the neural network, 4 nodes in, 4 nodes out, which means you have 16 weights. And you take the first input times the first weight, second input times the second weight, and so forth. And therefore, the computational complexity, if you use a single thread computing this, is actually n squared, right? Because the more nodes you have, exponentially the weight matrix increases. But the speed advantage really comes from being able to compute output nodes separately from each other and independently from each other. And that gets you to roughly linear, not perfectly linear, but O of n speed, right? That's a really good scaling for sizes of neural networks. Now, we are not that interested in those networks. We're interested in Markov brain networks. Markov brain networks have, and the ones I'm talking about, have a binary state vector. We have lots and lots of deterministic logic gates reading from those and then updating this binary state vector accordingly. Now, what about the computational complexity of a Markov brain in theory? Well, since all gates can, at least theoretically, independently of each other work, we technically have an O of const computational complexity. It doesn't matter how many logic gates you have, as long as you make sure that they're not overriding the outputs. You can scale arbitrarily large neural networks and still compute all of them in a constant compute time. So technically, we are down there at this green line and it doesn't really get faster. And then we're also using deterministic logic gates, which means that we can pretty much take advantage of the CPU hardware, which is made out of logic gates, at least theoretically, right? And the largest computer that we have right now is about 5 gigahertz fast, has about 60 billion transistors, which translates to about 10 billion logic gates, which means we could run a Markov brain, in theory at least, if we could use all of those transistors, as a size of 10 billion logic gates with 5 gigahertz of speed. That's amazingly fast and amazingly large as a neural network. Now, how does a Markov brain actually compare to the size of a natural brain? This is really, really a contentious story and really difficult to estimate. But 10 billion logic gates, which get you in the bottom right corner of this plot. However, it's not clear how one logic gate translates to one neuron. Maybe one gate is 1,000 neuron, maybe one gate is 1 neuron, maybe 1,000 gates is a neuron. When it's a 6 order of magnitude ballpark, it's not really clear where you are. But it would be quite a significant advantage if we'd be able to run much larger Markov brains than we can do this right now. Now, let's look at how we are currently evaluating Markov brains, right? We're not using a single logic gate on the CPU to update a single logic gate in the Markov brain. In fact, we're having a for loop that iterates over all gates, and then for each gate, we have a complicated memory lookup table. Yeah, if you think about the gates that you're using to support this maneuver, it's mind-bogglingly how many gates you're using to actually run even the smallest Markov brain. In fact, your keyboard controller has probably sufficiently many logic gates to run this thing faster, if you would be able to utilize them directly, than what we are doing between CPU and memory. Okay, so we are wasting transistors tremendously. So our computational complexity is probably somewhere O of ouch, right? If you think about how fast it could actually be. So, and then on top of that, we are not using backpropagation, we're using a genetic algorithm, which means we have populations of agents. Each agent has lots and lots of time steps that we have to evaluate them, and then we have to do this over many, many, many generations. So you see, wasting computational resources is incredibly terrible in this regard. So we would love to make this much faster. And there's crazy potential. And of course, the answer is just use an FPGA, right? FPGA is a field programmable gate array. It basically is a chip that has lots and lots of logic gates on it that are customizable or configurable such that they can actually implement arbitrary logic gate circuits. Cool technology, absolutely. And so what we would need to, for each generation, for each organism, translate the genome into a Markov brain and then into a bit stream. And the bit stream is actually what encodes the FPGA. And then there's an encoding step on the FPGA that has to happen. And then we can evaluate the brain in all-cons time. However, in terms of sort of, open science and being a really cool tool for everybody, we would love to have this entire process cross-platform, should run on all your systems. It should be fully transparent. Every step should be known. Otherwise, how do you really do science here? And then, of course, it should be free, right? So we can't really do anything about generations. We can't really do about populations, but we're really into, and then of course, these are all requirements that we pretty much pretend for now they're fixed. We have to have this. And then we're going to look at the translation step, and we're going to look at the encoding step. And then once we have all those things, we pretty much can show that, yes, we can run Markov brains in all-cons time. It's kind of the outline here. So let's look at the first one, translate genome into bitstream. Now, we have two major vendors here, AMD and Intel, making up 99.6% of all FPGAs available on the market. There are a couple of others, but these are the ones, right? Well, they don't make FPGAs for OS X. Well, I mean, you can plug in a configured FPGA into OS X infrastructure, but the design software and everything isn't made for that. And it's really usually a hassle. And yes, design software, we have Vivados and Quartus, Vivado and Quartus, and they're not running on OS X. So that means you can, even if you have a Verilog scheme, you cannot translate that into a bitstream on a Mac. It just doesn't work. Okay, that's a bummer for fully accessibility. Well, the software isn't free, and I have to say for all features, because they actually give you the free software, but you cannot take advantage of all options of the software. And secondly, the moment you're doing this in a business context, you actually have to pay a license fee. This is comparable with having to pay Google a fee every time you're running your TensorFlow library. That's the business scheme of FPGAs here. So that's not really cool. And then software is proprietary, which means we have no idea what the bit stream looks like that comes out of it. It's custom to the chips in order to hide lots and lots of that technology. I understand the reason for that, but it basically means that we have no clue what actually happens on the FPGA. And then lastly, this translation step is actually slow because you have to run this through their compilers. And even for small FPGA, it's fractions of seconds. And for larger FPGAs, we're looking at minutes. And for very large FPGAs, we're looking at the compile time of hours. So every time you want to run a Markov brain new, you have to reconfigure it, you have to go through this compile step, you have to waste a couple of seconds. Okay, so slow is probably the biggest problem. We ended up having exactly one option here. This is an FPGA from Lattice. I think that's Texas Instrument. It uses the Yosus toolchain, which is available on all systems. There's iStudio, which is a design studio that is available on all systems. And yes, this is an iStix 40HX1K. It has 1,280 logic cells, gates, and it has 12 megahertz and using a serial port over USB. And this is kind of probably the slowest way to communicate with the device. And all in all, it's a little faster than a Commodore 64 and much smaller. Okay, so technology 30 years ago. Compare this option with PyTorch and GPU hardware availability. Right? It's an insane difference. Okay, I have a new FPGA now, and this one probably got stuck in a canal somewhere. But yeah, we couldn't use this one. Anyway, what about programming the FPGA? Once you have the bit stream, what do? Well, in the olden days, and there are tons of different ways of doing this, by the way, but in the olden days, you burn fuses, which means you couldn't program your FPGA once. That's really not an option here, because you would need one FPGA for the Markov brain you want to test. Not really good. You have EEPROMs and EEPROMs, yeah, a little bit better, but still UV light or radiation and so forth, not usable. And then you end up with the two prominent technologies. You either have an SD card. Well, SD card is great, lots and lots of space on it, but well, you have to swap your SD card every time you want to recode your FPGA. Not a good way. And then you have SRAM and flash memory. Cool, right? That's persistent. If the electricity falls out, it's still programmed and so forth. Sounds really great. Yeah, but SRAM and flash memory, on most cases, is reprogrammable about 1 million times, maybe 10 million times if you have super high-end SRAM. But To give you an estimate, if you have a population of 100 agents and you have to do 10,000 generations to optimize them, you burned out your FPGA in one evolutionary run. Cool. Not an option. So translation from genome to bitstream is slow, and we have insufficient reprogrammability capabilities across the board. So yeah, screw this. That's pretty much what we decided. Being able to do a custom FPGA hardware accelerator for Markov brains, not an option with this current technology. Taking all our requirements into account. And then we thought about Inception, cool movie, but we can do something like that. Remember, FPGAs can be customized to run every logic gate configuration. So we should be able to take an FPGA to configure another chip that's fast and infinitely reprogrammable FPGA that models Markov brains. So that's where the Inception part comes in, so to speak, this nesting. And yeah, we came up with a field reprogrammable accelerator for gate logic. I had the idea of this acronym Fraggle, I think, a week ago, so it's not in the paper, but I think it's a good acronym here. And this Fraggle chip does two things. One part, the orange thing, is really just the communication control. It reads the data that comes from the CPU, recodes all the logic gates to work, and gets the inputs into Markov brain and puts the outputs of the Markov brain back to the CPU. And yeah, right now, UART read and UART write, which is the slowest possible form of communication. Of course, a different FPGA would be able to do this much faster, but this is the one we had. And then the blue part is really the Markov brain. When we tested this, first of all, this works. We can totally fine evaluate Markov brains on this FPGA. It works seamlessly, translation works fine, and so forth. And when we proved this, we found that yes, the Markov brain actually updates in a single clock cycle. This is as fast as the FPGA can do this, and it doesn't matter how large it is. I mean, with our constraints. So cool, works and is fast. But of course, I mentioned the limits, right? This one here, serial I/O port 12 megahertz. This is so slow that when you compare this to a normal run on a CPU, when you take MAPE, modular agent-based evolution framework, to evolve a Markov brain, takes about, for the task that we looked at, somewhere between 12 and 20 minutes to get perfect performance. When you try to do this on the FPGA, you have to wait 2 days. It's not an option. It's all wasted in the communication, not in the memory update. And then these 1,280 logic units that we had on this FPGA allowed us to make Markov brains of size 16. This is kind of the smallest Markov brain that we typically do. And you wonder, why are we wasting so many logic gates? What's going on? We should be able to run much larger brains here. So anyway, the current model that we have is very small and very slow. In the answer to this question, why we are wasting so many logic gates, has to do with addressability. Every logic gate in a Markov brain, at least in theory, should be able to read any output combination from the state vector. So you have this all-to-all connectivity between all your theoretical logic gates and the state vector. In an actual FPGA, this is solved by using what's called a switchboard. And the switchboard configuration is kind of the magic very often in the compile step of the bitstream. We can do this because we have to make our own configurable hardware, and we use, well, something like a demultiplexer. And demultiplexers have the disadvantage that the larger the address space becomes, the even larger the number of gates become. This grows squared or exponentially. So we are wasting an enormous amount of logic gates on the FPGA, doing nothing else than this addressing. That's where our sort of gate sink is. And if we would be able to actually use the switchboard, that would be much better, but here, not an option. Okay, so let's think of what's theoretically possible if we would not make a new chip. So if we take this technology to take an FPGA and make our own chip. The largest FPGA that we have is about 60 billion logic gates, which is a very large chip. Runs again about 2-3 gigahertz, quite fast. What we can do in this tool is to actually implement a Fraggle, this thing, which can run 65,536 logic gates and runs about 2 gigahertz, which is insanely large if you compare this to what we're doing, and quite fast. What you would need to do is you would need to take one of those, I think probably Intel chips, configure it on a PC, create the hardware on the PC, and pretty much end up with a ready-made box that's a hardware accelerator for Markov brains. We haven't done that, but that's the best we can do right now, unless we're making our own chip. Okay, so of course, For every detail I pointed out, there's an exceptional alternative solution. I talked with a lot of FPGA designers and manufacturers, and immediately you tell them one of the problems, they tell the solution to this one problem. You tell them the next problem, they tell you the solution to the next problem. Perfectly fine, but the problem is that we need to satisfy all requirements at the same time. And that's really kind of not working with this technology right now. So anyways, thank you, Tori Schossel, my co-author, and of course you for paying attention. Thank you so much.
Thanks very much for the presentation, Arend.
Yeah, thank you.
Has anyone got any questions? Any questions? I have a quick one. Have you actually spoken to a manufacturer?
Yeah, we spoke. I mean, in the beginning, we spoke with lots and lots of manufacturers, trying to find out how to do this much better. And yes, there are, for example, we'll give you a quote here. There are partially reconfigurable FPGAs. It's like, of course, yeah, if you want to reconfigure FPGA at runtime, you have to do this. It's like, okay, fine. How we do is, well, you take your Verilog system that you want to reconfigure, you run it through the software that's proprietary and costly, and then you get a subsection of the FPGA that you can reconfigure once you went through the pipeline. And the pipeline again is minutes of compile time. So that's typically sort of the crux of these situations.
I actually meant to get them to manufacture a new piece of hardware, a small run.
This is exactly what we're thinking about right now. It's probably the only step that we can do if we want to take full advantage of this. It would allow us to actually have a switchboard access, which will solve our connectivity problem, and then have a good I/O plug and playable. That would be really fantastic.
Okay, I see Klaus has a question as well, so I don't want to hold the spot.
Oh, hello. I'm super interested in FPGAs and stuff, but I'm completely newbie on that. So this is a very kind of trivial question, but let's say you build a prototype of what you're doing. How long would you expect the hardware to work before you had to like replace or repair or something like that?
That's a good question. For example, I mean, we tried this with the serial port. One issue is that your communication channels should be really in sync with each other. And we have a chip that runs roughly 12 megahertz, and then we have a CPU. My laptop computer runs roughly 3 gigahertz. I mean, that's not the speed in which really the serial communicator works, but you get out of sync at some point. So here, at some point after hours, suddenly your FPGA is out of sync with your serial port, and then you can't use it anymore. You need to set a reset signal and so forth, which is technology that we didn't build in there. If you have better communication hardware, this pretty much goes away. It becomes as robust as a typical what is it, PCI Express card or something like that. And I think FPGAs are high performance compute devices. They are very, very stable with this regard. I think if you shield them, they become even better because, of course, you know, they're sensitive to heat and all these things as well. Background radiation, very much like a GPU, I would suspect. The other thing that's actually quite important to understand is they're slower than an actual CPU, and as time loss is done to actually verify that the computations actually finish before the next gate has to do an update. So when they run with 2 gigahertz, they're more or less guaranteed that all updates of all gates, however they're connected, actually happen within that speed. So when we have a clock cycle of 2 gigahertz, that is the fastest speed you can do, and all gates actually finish at this. There are lots and lots of arguments when you have very large chips that sometimes you flip bits and you lose accuracy. And yes, that's technically something that you would need to build into your optimization process. And if you optimize your Markov brains on that device, they would come with that robustness coming from the noise that the device generates automatically.
All right.
Thanks, Aaron. Okay.
Thank you.
Thank you. I see also Timothy Atkinson has asked a question in the chat, except he's the next presenter. So maybe you could wait till after he's had his talk to get back to him. So the next presentation, thanks, Arend, for your presentation too. The next presentation is by Timothy Atkinson and Nihat Engin Toklu. It's on self-recognition as optimization. I'm assuming I can see Timothy's name in the list. Are you presenting, Timothy?
Hello. Yep.
Yeah.
Can you see my screen?
Yeah, we can see your Slack. Yep. Okay.
Oh, yeah.
Off you go.
Sorry. Okay. So, hello everyone. I'm Timothy Atkinson. I'm a research scientist at Nascence. I'm presenting a work done with my colleague Engin Toklu, which is on framing self-recognition as a optimization problem. So, a quick overview of what I'll be talking about. I'm first going to just have a quick overview on self-recognition and the idea of doing mirror testing. And then I'm going to move on to how we can kind of transition this idea to a general framework for doing things analogous to mirror testing for recurrent neural networks. And then I'm going to describe a simple experiment we did based on the classic mountain car environment and some results we got out of that. And then finally, I'm going to wrap things up with some conclusions. So the first thing we should probably talk about is self-awareness versus self-recognition. Self-awareness is a very, very big philosophical concept. Obviously, I'm not a philosopher. I'm not going to go particularly hard on that topic, but generally it's this idea of the ability to become the focus of one's own attention. It's a very, very big and complex idea. Self-recognition is a bit harder to pin down. It's generally physically observable evidence of self-awareness, like when you can see that an animal or a human has recognized themselves or recognized some sort of causality of their own actions or these kind of things. So self-recognition is the idea that we're tackling here. And the most familiar idea in self-recognition for most of you is probably the mirror test, which is used to detect self-recognition in animals, most famously first with chimpanzees. Since then, there's been experiments on orangutans, dolphins, elephants, magpies, a whole bunch more. If you go to the Wikipedia page, you can find lists upon lists of successful and unsuccessful mirror testing experiments. I'll talk about what the mirror test actually is in a moment, but it's worth pointing out here that it's generally criticized because it's too strong. It unnecessarily excludes some animals. A good example of this is dogs, because dogs don't really use visual stimuli too much. So they fail the mirror test, but they can do observable self-recognition through their smells, particularly smelling their own feces. They can actually recognize themselves there. So it's not generally criticized for not being evidence of self-recognition, but because it overly excludes animals from being considered self-recognizing. So the mirror test that you're most familiar with is the mark-die test. It's basically this idea that you take an animal, you sedate it, you mark it with a die where it's not directly visible to the animal if there's not a mirror there. And then you observe the animal, see how frequently they're touching the dyed area. Then you place a mirror in front of it, let them interact with the mirror for a moment, and then again observe the animal and see how frequently they're touching the die. And the basic idea is that once the mirror is there and they touch the die more frequently, they're using the mirror to see that there is dye on themselves, and then they're touching it more frequently, and therefore they have an internalized notion of self because they've recognized themselves in the mirror. So when you actually break down the animal's behavior, it's normally something like this. So first there's a social response, like it would when there's another member of its species. Then it moves on to some sort of mirror inspection, trying to establish what the mirror is, because most animals aren't familiar with mirrors. And then after that, you see this repetitive mirror testing behavior, actually confirming that what they are seeing is themselves. And then finally, you get this self-directed behavior where they're using the fact that there is the mirror to achieve something else. So in that case, actually inspecting the dyed area they've seen on themselves in the mirror, and they are directing that using the mirror image. What we would argue is the most interesting part of this is the repetitive mirror testing behavior and the self-directed behavior. So really it's these things that we're going to focus on as what underlie the actual behaviors we want to observe from the mirror test. I mean, obviously the social response and the mirror inspection are interesting, but they're also products of the fact that the animal exists in some sort of social hierarchy and exists in the 3D world where there are mirrors you can look behind, and that makes it a lot more complicated to try and move towards a simulated analogy of the mirror test. So these third and fourth points are the ones that we're most interested in. So it turns out you can build quite a general framework out of the box, and you can apply it to almost any existing reinforcement learning environment. And essentially what you do is you have an environment, and then you place the agent in this environment, and in one of two cases. So in the first case, it observes itself, and in the second case, it observes some other. So exactly the same observation space, but the actual environment is being controlled by a different agent, not the self. And then you do not tell the agent which of the two scenarios it's in. So you then reward it depending on the two cases. So if it's observing itself, it solves one task. If it's observing the other, you reward it for another task. And you make sure that these tasks are mutually exclusive. So maximizing one of the tasks should be minimizing the other task. And obviously, because in the second case, you're not actually letting the agent see itself, you should probably try and make sure that the second task can be solved without observations, such as something as simple as standing still, for example. And then what you can hopefully see here is that this basically induces the two things that I had on this slide, this repetitive mirror testing behavior and then self-directed behavior. Because the agent is not told which of the two cases it's in, it has to do repetitive mirror testing to establish which of the two cases it's in so that it can maximize the expected reward. And then once it has established which of the two cases it is in, it then has to do self-directed behavior in the case of task A to eventually solve task A. So it's maybe a bit easier to then see how this works for an actual reinforcement learning environment. So we took the classic mountain car environment, and the left-hand side you can see here is the classic environment. So you have a car that's on a track, it has a position and a velocity. The agent is controlling it by applying acceleration to it, and it has to move from a start position, which is down at the bottom of the hill, and then it has to reach this flag at the top, X1 fin. So, and generally to solve it, it has to do some sort of oscillating behavior to get out of the lower hill. So it's slightly more complex. So if you just took the left-hand side, that's the classic mountain car environment. And then if you add in this right-hand side, you have this second agent, and it's only seeing the left-hand side. So that's why we're using this analogy of a one-way mirror. So the first agent sees in a mirror, sees itself, has to direct to reach the target. The second agent starts at the origin and is rewarded for staying at the origin, X2 fin. So that's the task B we talked about, where you really don't want to make too complex a task in the case where the agent is not seeing itself. And you can see in the details of the paper, this is nice because we actually find a strict threshold where you can only surpass this episodic reward if you go past, if you achieve self-recognition. So that's a nice little property. So with that in mind, we set up a little experiment. We used a 64 neuron RNN. We used initially, the RNN has a randomized hidden state, which lets the agent take pseudo-random actions. We then frame this as a policy optimization problem, use separable natural evolution strategies with the ClipUp optimizer, and then run it with a large population and a large number of generations. We did this 10 times in two different scenarios. So the first was co-evolving two policies, which means that you have a policy for each of the tasks, which basically reduces to just solving each of the tasks independently. It's very easily solved. And then when you evolve a single policy to try and maximize reward in both environments, it becomes much, much harder. But the critical thing we observed was it is possible. It takes significantly more generations to solve, but we can train a self-recognizing agent in the scenario we described using existing evolutionary computation techniques. So that's the experiment we carried out. I have some videos of trained agents here. So on the top, you can see what happens when you just evolve a policy for each environment. So you can see the traditional mountain car behavior of an oscillation, go up the hill, and in the other case, stay at the bottom. And then in the bottom half, you can see a self-recognizing agent. So what's actually happening here is the same RNN has been cloned. And on the left-hand side, it's controlling the car to get to the top, and it is observing itself. And on the right-hand side, it's controlling the car that stays at the bottom, but it's observing the left-hand side and it's trying to stay at the bottom. So it's exactly the same RNN with exactly the same policy weights, but it is using the mirror to do some sort of mirror inspection, mirror testing, and then self-directed behavior to solve one of the two tasks. And if you actually observe on the right-hand side, the policy that was trained just to solve the right-hand side problem versus the policy that has to solve both problems, you can actually observe in the bottom right some gentle mirror testing behavior where it does these little oscillations, generally at the same time that the left-hand side is doing, and it's actually establishing whether it is controlling the environment that it is seeing. And it determines that it's not, so it stays at the origin point. And then if I switch back. So to wrap that up, we've focusing on analogies of the mirror test as evidence for artificial self-recognition. We've identified mirror testing and self-directed behavior as the key elements that we want to see. We've given a general template for how you can create mirror tests out of existing RL environments. We've applied this template to the classic mountain car task, and then we've demonstrated that you can actually learn self-recognizing agents using existing evolutionary algorithm techniques. We should stress we're not claiming that these 64 neuron RNNs are self-aware. We don't have any particular claims on the bigger consequences of that. If anything, it's maybe a more criticism of the validity of the mirror test as evidence of self-awareness, even though it definitely is evidence of self-recognition. We would also stress that we noticed this giant increase in difficulty when actually training an agent to self-recognize. And this maybe makes a template for generally producing more challenging, intrinsically hierarchical reinforcement learning problems out of existing simple problems. So taking, for example, a humanoid running task and applying the template we've used could make it significantly more challenging because of the pre-requirement to establish self-recognition without falling over, which is a requirement of a humanoid running task. And that is everything I have to say. So thank you for listening. I would just like to point people towards the fact that on 1st of August, our company will be releasing an evolutionary algorithm library built on top of PyTorch. It's incredibly powerful. And if you would like to know anything more about it, just contact me and I can sign you up for beta testing or anything that you like. So thank you very much for listening. If there are any questions, I'm happy to answer them.
Thanks very much, Timothy. Actually, Klaus has already entered a question, so I won't. I'll go straight to Klaus. It says, referring back to the criticism of the mirror task in the beginning of the talk, do you know of variations of the mirror test for non-visual or not so visual animals?
The main one that I'm aware of is one that's been used for dogs, which is that you can observe that they have a different reaction. They inspect their own feces significantly less than the feces of other animals, which is arguably is not a visual mirror test, but they are demonstrating some sort of self-recognition by saying this feces is caused by my own actions, it's mine, it's therefore less interesting than what I can find out from another animal.
So it sounds, if that's the criterion for the test, then wouldn't, for instance, a social insect swarm that rejects a foreign insect be passing the same test?
Well, so the individual insect probably isn't, right? Because the individual insect
it's recognizing itself.
But it's recognizing that
itself.
Yeah, I mean, I'm not aware of any particular literature or discourse on that, but based on my understanding, it does seem like the swarm has some sort of notion of self versus other.
Yeah, just interesting to think. It reminds me very much of an immune system, right?
Yeah,
it's a kind of a mechanical.
I mean, it gets a little bit more. It gets a little bit more complex when it's one of these distributed systems to actually make solid claims, I think.
Tricky.
It's certainly
right, any
something
other
that I have less familiar with.
That's all right. Is there any other question? James has one. If it's really quick, James.
Yes, really quick. Just wanted to highlight that when we were talking about the idea of a swarm passing mirror tests or self-recognizing, there was actually a project mentioned by Thomas Schmickel yesterday called Kokoro, where that was aimed to be one of the objectives was to have a swarm underwater being able to recognize itself using the mirror test. And like you say, the immune system is absolutely one of those systems which some believe to be self-aware, but as I'm sure we're all aware, as soon as we get into something like self-aware, it's like talking about cognition, and everyone has their own definition, their own opinion. So it's best.
Yeah, thanks, John.
Well, I unfortunately had to miss that, so I'll probably try and catch that now.
It should have been recorded, I'm sure. You'll be able to watch it.
Yeah, I'll try and catch the
Quite
recording.
a project, but it was just because Thomas was part of that team, and so he just mentioned it in the intro, that's all. But if you drop me an email, I can forward you to papers and things.
Yeah,
Great.
sure, thank you.
Thanks. Thanks again, Timothy, for the presentation. That's great. Let's move on to the last one for the session. Shuto Kuriyama, Wataru Noguchi, Hiroyuki Izuka, my mistake, Keisuke Suzuki, and Masahito Yamamoto. It's a lot of authors for this paper, which is terrific to see the collaboration on gradient climbing neural cellular automata.
Can you see it?
Yeah.
Okay, cool.
Shuto there.
Okay,
thanks. Go ahead.
Thank you for introducing. My name is Shuto Kuriyama, and I'm gonna explain our research, gradient climbing on neural cellular automata. The difference between life forms and non-life forms, as well as fundamental features of life form, have been researcher interests both in biology and computer science. And as a fundamental feature of life forms, we know self-generation and then self-maintenance proposed in the autophagy theory by Varela. We also know self-movement that occurs when a life form moves in response to an environmental gradient or difference. It's called chemotaxis. And there are several computational simulations that simulate these fundamental features. First, the model that simulates self-generation and self-maintenance and self-movement was proposed by Suzuki and Ikegami in 2009, and they extended the autopoiesis theory by adding self-movement on 2D cell systems. And the agent climbs up its environmental gradient while maintaining its self-boundary. However, this model lacks in the complexity of the shape of generated agent. Next, the model that simulates self-generation and self-maintenance was proposed by Modo Vincev in 2020. The model is called neural cellular automata. And the model generates a complex shape from a single cell and then maintains the shape by cells' local interactions. And also the neural network is used for a CA update rule so that the update rule is learnable. However, the self-movement, which is the fundamental feature of life, was not simulated on the model. Therefore, with our research, we aim to build a model that simulates self-generation and self-movement, self-maintenance and self-movement with complex shapes. We added self-movement simulation to the neural cellular automata model so that the agent climbs up an environmental gradient. Before explaining our model, I'm going to first explain neural cellular automata model. And so the model has 2D cellular automata, and each cell state is a 16-dimensional continuous vector. And so that there are 16 channels for cell states, and the first 4 channels represent RGB image. And the model runs deep-wise convolution 2D and input the result to the 1-by-1 convolution neural network, and then output an incremental update rule. And then after input the result from neural network to stochastic function, and then add the result to the previous states, cell states, and then now input them to a live function and output update cell states. And the stochastic function adds randomness to the update and prevents each cell states from updating synchronously. And for the overall architecture, The model inputs cell states to update function, which I described in the previous slides, and then update them recurrently. And after calculate the loss between the RGB channels of target generated pattern and then target image, and then back propagate the loss to the neural network in update function. And also the model has a structure called pool that store multiple cell states. And the model picks up several cell states from pool and then update them recurrently. And then after the update, the model replaces cell states sample from the pool with those updated. This allows the model to generate target pattern from various cell states. As a result, the model can generate a complex shape from a single cell and then maintain the shapes. Next, I'm going to explain our research model. So we added chemotaxis, self-movement, to neural cellular automata. And in order to do that, we added 17th channel that represent gradient, environmental gradient. And the agent moves towards 4 directions, left, right, up, and then down, and in response to environmental gradient. And for the gradient, we use 5 types of gradients, and the 0 gradient has the same value all over the grids, and the directed gradient has the value increasing towards the direction of the gradient. And the model generates each type of gradient randomly. And for CA update, the model sets gradient at the 17th channel. And then apart from this, all the calculations same as neurocellular automata, as I explained before. And we use different target images for different gradients. And for the 0 gradient, we use the same target image that was used for neurocellular automata. And we, for the directed gradients, we use a target image that has an agent shifted towards the direction of the gradient. And we also use the pool structure. So the model picks up several cells, cell states from the pool, and then generate 5 types of gradients, and then set them to the multiple cell states, and then update them. And after the update, since there are several cell states that has an agent shifted to the gradient direction, we shift them back before putting them back to the pool. And the total loss function, was calculated according to the loss equation below. As a result, we've tested our model to the 5 types of gradients, and with the 0 gradient, the model can generate the agent, and the agent climbs up those 4 types of directed gradients. Next, we've tested our model to the per-tube gradients to validate the robustness of self-maintenance and self-movement. And we added a noise to each directed gradients. And as a result, the agent climbs up those directed gradients. Next, we tested a model to gradients that was not used for training. And with the hill gradient, we expect the agent to stay in the center. And with the valley gradient, we expect the model to make a decision which direction to go between left and right. And as a result, with the hill gradient, the agent shrunk its body towards center and stay in the center. With the valley gradient, the agent expanded its body towards both left and right, and after moved towards right. We also tested the model with the valley gradient with the lowest vertical line shift to the right. With the one shift to the right by 6 pixels, the agent didn't go neither left and right, and instead it moves towards bottom. Since there was no gradient to the bottom, we think that it was caused by internal dynamics of the agent. And the one shifted to the right with by 7 pixels, the the agent expand its body, and then after move towards left. A summary, we proposed a model that can simulate self-generation, self-maintenance, and self-movement with a complex shape. Regarding self-generation, the model can generate a complex shape as seen with the neural cell automata. And for self-maintenance and self-movement, the model showed a gradient climbing behavior, responding to the 4 types of gradients: an unseen gradient, which is valley gradient and hill gradient, and a perturbed gradients, while maintain the shape of the agent. Additionally, after we submit our paper to the conference, we found the research that did similar thing with us. It's called goal-guided neural cell automata, learning to control self-organizing system, by Sudhakaran and others. And this was published on the workshop of ICLR on 29th of April in 2022. And the model is quite similar to ours, but the difference is that they used a mask instead of a gradient. So with their model, they input the information of direction to the goal encoder, which is composed of a neural network, and then output a mask, and then add the mask to the cell states. And then after, they run the same simulation, the same calculation as the neural cell automata. Because of that, in the training process, the model backpropagate the loss to the neural network in the update function, as well as the neural network in the goal encoder. This is the end of my presentation, and thank you for the listening.
Thanks very much, Shuto.
Thank you.
Anyone got any questions? You can speak or type as you prefer. No questions. Is this part of your PhD, Shuto, or something?
This was my bachelor, like a thesis project.
Okay.
Yeah.
Thank you. Any questions? It looks like you've got off easy.
Okay.
Great. Well, thanks again very much, Shuto, and thanks everyone for attending the session. It looks like we're going to finish 2 minutes early. Okay. Thanks. Please, everyone, thank all of the speakers. And I look forward to seeing you in the next session.
Great. Thank you, Alan. Yeah, hi, Arend. Yeah, the new chair for the upcoming session will be Arend.
Yes, thank you so much. I'm the new
You're
chair.
welcome.
We have 5 interesting talks ahead of us. The first 3 are 20-minute time slots, including discussion, and the other 2 are 15-minute time slots, including the discussion. Yeah, I mean, we are close to 12:30, so I guess we just start with our first talk, Ethics on Artificial Life, the Moral Status of Life as it Could Be, from Olaf Witkowski and Eric Schwitzgebel. I hope I'm pronouncing this right. Who is going to present of you two?
Thank you, Arend. Let me just, I'm trying to connect, so let's maybe give ourselves 1 more minute so people...
Sure, we have time.
Then I try to connect to Zoom with my computer here, which is non-trivial.
Yeah, all is fine, we have time.
All right.
Yeah, we can see your screen share.
Amazing. All right, let me wait for a few more seconds in case maybe people want to join.
This is in the online time so much easier. You just click, 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 and you're in a room. So, but it means that you cannot pick up a coffee on your way there. That's the downside.
That's right. I guess I mentioned that on this topic we'll have a. Actually, I mentioned it in the talk, so never mind. Okay, 11:30. Try start.
Yeah, please take it away, and I'll send you a text message if you run over.
Sounds good. All right. Hi, everyone. Welcome to this talk about ethics of artificial life. And I'll talk about the moral status of life as it could be. This collaboration with Eric Fritz-Gobel. Here we start. We're organizing, Eric and I, also a workshop on the same topic because we think it's very valuable to think and get feedback from the community. So if you are free tomorrow morning, a bit earlier than this, please join us. We'll have nice speakers here listed on the right. And we need your feedback, so please come. So first, why, ethics, let me start the timer here, of artificial life. Artificial life is dealing with synthetic systems that may come either now or in the future deserving of moral consideration to some extent. And them being very different from human beings shouldn't really preclude them from having rights or moral consideration. We also, they owe us their existence, of course, in most cases, and that shouldn't prevent either them from having rights. And on the contrary, the creators of ALife systems may have some extra obligation because of this creation, a bit like pet owners owe special obligation to their pet or parents to their child. And I think there's a huge field of application and sort of responsibility for us ALifers to think about those questions. So here we go. If artificial life is larger than life, then perhaps the ethics of ALife beings should be larger than human ethics. Everything starts with human rights because everybody has a sort of intuition that humans, just because they're humans, we have rights. And those are fundamental, but they don't have to be. It can be also construed in a more general sense. And we have this intuition that we deserve a very high degree of this moral considerability. And other entities are currently very far from sharing any comparable status. Now, it's kind of controversial, but obviously, what is not is that we don't all have rights, and we still have a lot of work to do on that. And I don't want to minimize any of that by giving this talk. We do feel that this is very important as well, but a different, slightly different question in parallel. Humans, although they deserve those natural rights, we feel, are not so unique. And there is this anthropocentric sense that humans have long been thought as unique in different aspects, but two of them are still very much alive. And one is intelligence, right? We're so intelligent as humans, and we're so much more conscious. We have this special feature of consciousness, and we're not sure that others have it. And consciousness has been largely viewed recently. It has been sort of revealed that different aspects that were considered unique were not so unique anymore. And I won't go into details of this review that we do in the paper. But right now, those mechanisms are recognized as potentially existing in non-human beings as well. And if human beings have no unique capacities that ground this high moral status, then they might also not in principle be uniquely deserving of those highest moral status that we think for them. Animal rights, animals are the next candidates in line, essentially, non-human animals, that is. And some of them do enjoy some rights already. Those are very limited. Many jurisdictions do award prison sentences for abuses of pets. And the UK have recently extended that to cephalopods and crustaceans. There are different limit cases. There are different types of rights, of course. There's copyrights. Do animals have that? There's a lot related to food, what we should be allowed to eat, how we should be allowed to eat it, and so on. This is a very complex question, but it's much more evolved in terms of debate and jurisdiction than any artificial entities, which may be considered as well as deserving rights. And this is where the controversy really explodes. I guess this moral considerability may be considered for artificial entities, but it really differs from the places, the fields where you are in the movies you're watching. depending on that, you might have a completely different view, widely different point of view in terms of who deserves what. In Japan, for example, there is a sort of empathy towards artificial beings, especially robots, things that you can interact with. And maybe it comes also from some kind of animism, different influences, not only religion, but also culture, also sort of different respect to different things. I think it's very interesting to study. Now, there is a sort of agreement that some of the entities may potentially deserve in the future in some degree, some consideration. And there are a lot of ifs you see there, which is very tricky to entangle, and maybe it shouldn't be remaining at this iffy stage. All right, so there's a similarity between beings that augments that. I'll touch on that later. The examples here are pretty wide, right? In Japan, you can marry a video game. You can, there's a bunch of things. I see that you see that I put Lambda in here. That leads to us talking a little bit more about AI. Won't address that controversy that was kind of misplaced in the news. But yeah, a few quotes here that are saying, right from Hans Moravec, Yoshua Bengio, that AI is not so intelligent yet, right? So especially it's very narrow, plays very well at chess, at Go, and so on. But as soon as you reach more general, it's not so intelligent. Again, that speaks to the seeming uniqueness of things. Human intelligence may not be as general or as high level as we widely think or used to think it to be. We tend to use human intelligence as a basis for reasoning about others, less familiar phenomena of intelligence, though. And this is a huge bias that biases also our theories on moral consideration. And we have to be conscious of that. And yet AI has a huge potential, and other artificial beings might have huge potentials in terms of intelligences that we don't really think about. And maybe in the future, those will become very, very obvious. All right, so I think artificial life goes beyond those wild differences in AI. And I think people in the room will, in general, agree with me naturally. But yeah, I'm just giving you here a few examples from cyborgs, robots, hybrids, right? Like those hybrids are very important. So ALife is really about the synthesizing and simulating living systems, right? And a large set of those fundamental problems, including the lack of formal definition, is something we deal on a daily basis with. So we have some sort of grounds in interdisciplinarity that may contribute to this kind of debate, which is why we're organizing this workshop exactly. But a particular point I want to insist on here, I want to insist on everything we mentioned in the paper, but it's hybrid systems. And I think those are very interesting because they won't happen in the remote future. They're happening now very soon because we are mixing naturally living and non-living in our technologies, and also different things like different categories like external designers, mixture of mechanical, electrical, chemical, biological components within. And that creates a very wide, multidimensional space to explore different properties of artificial life, as opposed to just being alive or not alive, being moral, considerable or not, right? So this is not a black and white. It's not only even gray. It's many, many shades of dimensions that we have to consider, and that's the approach that we are suggesting here. Moral considerability is related to often suffering, being morally bad or not, on account that the entity itself, and regardless of the consequences on other beings, right? So this is something that has different approaches in the literature, either utilitarian, you have an equation of this is good or bad, we're going to make this cut, right? This is leading to trolley problems, and etc. We won't go into that. There's individual rights-based. What I'm insisting on this slide is just a very simple message, is that in ALife, we already know about this, but we can use our tools to study it more. Essentially, these approaches that understand already how life can merge, divide, the boundaries being vague, permeable, this is something we have sort of an understanding about. This may contribute to those debates in very direct ways. All right, don't have that much time. I'm suggesting 5 criteria. This is a huge simplification, so don't hit me. Well, yeah, I'm morally considerable, so you shouldn't, but also I'm a nice person. Embodiment, consciousness, cognition, social, and group entities, extended mind. So those are 5 important criteria that we think are worth mentioning. So in our review, the first one, embodiment, first, so similarity of physical embodiment should probably not be a determining factor for moral considerability, we think. In our view, we should sort of put this aside. And of course, the cognition or sentience of an entity may depend on its medium, but we shouldn't use that one as a decision directly, right? So this embodiment equals this kind of consideration. That's a no-no. Most current theories focus on psychological or social properties instead. Second is consciousness. I could spend so much time on this, and yet, yeah, I did read a lot. Still don't understand exactly what everyone means. But what we gain as an understanding is having consciousness might be necessary for moral considerability, or some degree of it, or it might be sufficient, or a bit of both. So the most liberal views might involve some kind of denying that the mere existence of this consciousness is sufficient for a high level of moral considerability. Sentience, different, but yeah, in a more narrow sense, there's something a bit different. So there is existing work, essentially, maybe I'll insist on that, about fish pain. And often, well, you can't ask a fish whether it feels pain or not, right? So you have to test its behavioral response. So that has been the major approach. And of course, that comes with its own issues. This brings me to our next point, to save time and speed up a little bit because I'm not speaking fast enough. All right, there is cognition. Cognition seems to be very important, but essentially, presumably, the capacity to add numbers, for example, is something your calculator does very well, or things that the computer does very well may not be directly linked to how much moral consideration you give to it, right? You may buy another computer, repair it, I don't know. This should not be linked. Maybe a more direct example is infants, babies that that are deserving of moral consideration and that cannot add numbers for to any extent until a certain age. All right, so there are different architectures that may allow, right? So there are minimal conditions, arguments, and so on. There are cases of Lupton syndrome and so on that are interesting also in this case. All right, maybe not too much insisting on examples. Our fourth one is about social attribution. It's very interesting that being intersubjectively attributed moral status within a group might be sufficient for moral considerability in certain cases. I have a funny case because it's a sort of, I guess, inner private joke at ALife that we talk about topaz, right? Those social entities that are essentially imaginary friends that because you care about them, maybe other people should care about too, otherwise they would hurt you. And maybe a more direct example is, I'll jump into that in a second, with the prosthetics or extensions of our mind. But before that, there is the example of the uncanny valley. So there may be some biases towards evaluating very highly certain entities or very moral choices, depending on the aspect or what we think, how artificial we think the entity is. And this is a very important aspect. It's not the only bias that we will have, and we have to be very careful about those biases. All right, last one is this extended mind thing. Maybe the example of someone stealing your smartphone or hitting it with a hammer might hurt you directly, and it happened to me, and it certainly hurts. So maybe we should consider it as such. So that seems important. Just jumping into discussion, so we have time for questions. I hope there will be some. Oh, 15 minutes have passed. Oh no, I'm at 14 here. All right, let me take a minute to conclude then. All right, just a few examples here, but essentially there may be some practical applications. This Hitchbot, right, that was tortured on its way to the US from Canada. The issue may need to be studied in advance. So we come up with policies whenever the problem occurs. It's not now, right? We don't need to know now, but it's really good to study it in advance, especially for our field. I think the study of hybrid systems, again, is very important, and there are limits for cases there. Yeah, thank you for questions coming in. All right, maybe I skip on that just to give a final remark. So yeah, very important to think about this, but even more important to think about universal human rights right now. And I don't want to minimize any of that. And I want to invite you guys again tomorrow for the workshop, and I welcome questions maybe quickly now. Thank you.
Cool, thank you, Olaf. I think Steen raised his hand, and then we have.
Yeah, hi Steen.
Hey, hello, Olaf. So yeah, this is certainly a very interesting topic, and it was a good sort of a tour de force. I have a question. What do you think about all these sort of semi-autonomous systems that's being deployed? I mean, one extreme is drones that are used in war, and also, but also quote unquote benign systems that are used to decide who's getting social benefits or not. I mean, how much of the moral responsibility is distributed between the creator, the user, and so forth and so on? I think these are really pertinent questions. I think your issues are sort of more futuristic sci-fi issues. So I'd like to hear what you think about here.
Yeah, I have a remark. I sort of skipped over it, but we have not only responsibility to the entities, but of course to the effect that they give. So that's the same with your pet or your children. If your pets bite another child, whatever, well, you are very highly partly responsible, right? And yeah, this is very important consideration as well that we should discuss. I hope we can touch on that tomorrow. Thanks, Steve.
So which one is that? Which of your numbers?
Sorry, numbers for?
You said there were five areas. So which of the areas?
Oh, I think that would be, that would be on one of the two last ones. The social and I guess shared extended mind.
Okay, thank you.
Cool. We have lots of questions in the chat. Do you maybe want to take the first one there, and then?
Sure. So Ettore I think that social attribution is effectively the way our civilization attributes morality, but it does not explain how we get to such agreements. Is in your view that ABCE form D, or do you see D as independent? So yeah, for sure. This social attribution is very important. I think they all interact with each other, to be honest, right? This one is the one that is usually forgotten, and we added that one in. Usually you forget that someone considering something as important is part of your care circle and should be. So it should be within your moral attribution sphere as well. Yeah, maybe that's for one quick answer.
And the last one maybe from Seth.
Yeah, maybe from Seth. Alan said something about environmental processes that I find very important. Let me find Seth's question. Oh yeah, similar. What do you think of Kaufman's reinvention of the sacred as an approach to a new ALife ethics morality? So I haven't read that, I confess. I just addressed the environmental point as being very important, and I guess we are part of that system. As in, so maybe we create ALife, and they are part of our system. And we are part of the larger system, which we have to be conscious of. And this is part, I think, of the same kind of conversation in terms of layering those systems and understanding how they are morally responsible for each other. There is stuff that is, our society is responsible for us as well to an extent. And we tend to forget that when we imprison people because they did something wrong. Okay, I'll leave it at that. But I think this is an important question indeed. Thank you for all questions. I don't want to take too much time. But please come again tomorrow, and we'll have 3 hours to talk about this.
Yeah, sorry, Alan, I didn't want to cut you off. I overlooked your comment. You were earlier. Maybe yes, the tutorial or the workshop is the right place. Let's go on with our next talk. Ana Rubio Deniz, I hope I pronounced this right at all, on Q learning for real-time control of heterogeneous microagent collective.
Yeah. Hi, thank you. Let me just get my screen up. hopefully you can all see that.
Yes, we can see that.
Yep, so I'm going to be presenting about our work on working on controlling heterogeneous microagent collectives. Specifically, this project looked at using Q-learning, so using the reinforcement learning for this control. But I'm going to talk a little bit more generally about the project that led up to it and the hardware that we developed to do this, and then sort of get into that at the end. So I work at the University of Bristol. I work between Thomas Gorachowski and Sabine Hallett's labs. So that's a swarm lab and a biocomputation lab. And some of the work in this project was done by a master's student, Leah Matu. So talking about control at the micro scale, why do we want to control agents that we find at these scales? We can think about all sorts of different agents that are really useful to us. So the ability to control mammalian cells and bacteria. And then on the inorganic side, we have things like microparticles and micromotors. So thinking about things like cancer treatment, wound healing, control of biofilms, the use of micromotors for cleaning plastics out of the ocean. These are all just a few of the examples of these kind of micro manipulation projects that are going on across science. But the big issue that we obviously face controlling these small scale agents is they are relatively simple. They're hard to program. They have limited programmability, and they also tend to have to work in very large numbers. You rarely will have one bacterial cell, one piece of algae, one micro or nanoparticle. They work in very large numbers. So developing control strategies for that is obviously challenging. And so the way that we've been working on that is by using light as a control mechanism. So obviously there's a whole host of different ways that you can do this. A lot of people use, for example, magnetism, electric fields. I think light has its own benefits, some of which I've listed here. So you have the capability for independent control. So you can have two agents at different points in space, and given the right controller mechanism, you're able to give them two completely separate inputs, which obviously for something like, for example, magnetic fields, you can't do because it's obviously coupled. On the same token, you can also have the capability for orthogonal control, so using different wavelengths to interact with things in different ways. A lot of things are responsive to light at different wavelengths. Relatively low agent damage and a large range of responses. And by that, I mean all these examples everyone on screen. So you can, depending on the kind of medium you're working with, light can be used to generate heat, to generate currents, for chemical transformation, catalytic reactions, and for more organic processes such as phototaxis or protein activation. So thinking about more things like optogenetics. So light's a really powerful tool at these scales. When you get down to these very small micro nano scales, it does a whole host of things that obviously on the macro scale we don't necessarily think about. But the issue is obviously more so, how do you then deliver that in a way that's effective, in a way that allows you to still interact with your system? And these are just a few examples from some other people's work of some really cool light-based micro control work projects that are going on. So things like controlling polymer microbots with traveling light waves, using micromotors to induce collective behavior, again, often for things such as cleaning microplastics out of water, and more biological systems. So biohybrid microbot that you can steer using light illumination. And the shaping of algae and bacteria. So you can see you've got algae shaped into a nice shape over there. And then we have E. coli that was actually used to pattern faces. So you've got there the Mona Lisa. I think they also did Albert Einstein. So some really cool stuff that you can do. And so for our project, what we kind of specifically wanted was to develop a closed loop system that would allow you to have whatever sort of generic microsystem you have, something like responsive, so some of the things I've already spoken about. Have it so that you could deliver these kind of light patterns that you could shape in real time, obviously linked back then with real-time image analysis, and those two kind of coupled so that you can shape the light pattern around the evolving dynamics of the system. So for example, where the agents are moving, what the kind of density is, basically based on the behavior of the system, you can then create these light patterns with which to interact with the system. And the way that I've developed to do this is using the DOME platform. That stands for Dynamic Optical Microenvironment. So again, this idea of shaping our environment around our system. And there's a picture there, I think that's actually a bit of an early prototype, but, but there's a picture that gives you an idea of what it sort of looks like. There's a slightly more up-to-date version. So it's kind of a desktop device that can sit and plug into a monitor, or you can control remotely from a laptop or computer. You have a projection module that sits at the top, so it uses a digital light projector to create these illumination patterns on your sample, and then an imaging module that obviously allows you to do that imaging to link back. And it all runs on, all the computational modules are based on Raspberry Pis to keep it kind of low cost and accessible. And I've got the optical setup here as well. So you have your projector, goes through the condenser lens onto your sample. So you get these really tiny light patterns, and you're able to image through a series of lenses and camera, and that all like connects back for your real-time feedback. You can then calibrate the two together. So the way that works is by creating a projection image. So I do a 2x2 grid. You can then obviously read that in through the camera and figure out the matrix transformation you need, so that when you're then running your algorithm, you can shape these patterns and sort of feedback from the camera images what kind of projector image you need. And yeah, I mentioned it's kind of open source and low cost. That was really important. We wanted to develop something that not only could we produce a few of, and right now actually there's a few different projects that are going on that are all working on these. So we've had a few of them now that we've made. But also obviously for anyone else, if they were interested in building something like that. So all the main kind of structural components are 3D printed out of just standard PLA, so something that most people in sort of university and research institutions would have access to. They're all available online, and then all the other sort of parts, all the electronic, all the computational, all the mechanical components, again, relatively low cost. So the whole thing you can build sort of depending on what kind of lenses and stuff you want for under £700. And again, the parts and the kind of the basic calibration code and stuff is all available online. So some of the basic features. So in the model that was used for this work, the standard wavelengths in the projector we use, we're able to illuminate in blue, green, and red. So kind of going back to the idea of orthogonal control that I mentioned previously. But actually in a more recent project we've been working on, we've actually managed to switch out those light sources. So we're now able to customize that. And actually a project that we're doing now at the moment, which is looking more at wound healing and collective cell migration, we're using near UV wavelengths for that kind of activation. The latency scales with the camera resolution, but for the work that's presented here, we were using a 920 by 1088 resolution, and we had a latency of 0.25 seconds, which for everything that kind of we were working on seemed to work fine. Obviously, it might be that if you had very, very fast moving agents, that might be something that you would want to scale down the resolution or look at a slightly higher power computer. But for everything that we did here, it seemed to work fine. And in terms of the resolution of the projection, so at the moment, smallest resolution, so if you have 1 pixel on a projector, that maps to a 30 by 30 micron space in real terms. So that's about the size of a mammalian cell, like a skin cell or something. So taking it back a second, that's kind of the platform presented, a quick tour of the dome. I'm just going to mention now a little bit about sort of some of the motivation for this kind of work on this project specifically, and that's thinking about building blocks towards swarm control. So swarm behavior can be a whole host of different things. I've kind of got a bit of a breakdown here. Things like pattern formation, aggregation, coordinated motion. So we were thinking about this, and what we really wanted was to have a way where the kind of original idea of the dome was to create a small scale swarm arena. So we actually have a large version of this at our lab, but we wanted to create a small version where we can put in our micro agents, and we can start to play with inducing collective behaviors into them. So we kind of really thought about how to break that down and how to do that when you have these relatively simple agents. So assuming you just have an agent that is in some way light responsive, how do you start to build up towards these behaviors? And we kind of broke it down into 3 categories for now. So signaling, stigmergy, and motion control. Signaling being a direct form of communication, so more the way that maybe we would exchange information by kind of coming into close contact and directly exchanging a signal. versus stigmergy, which is more akin to ant pheromone deposition. So moving around your environment, depositing signals or information for other agents to pick up at a later date and sort of communicating in that way. So that would kind of usually be an either or. So there's a kind of two different ways that you can communicate, which obviously is very important for collective behavior. And then we had motion control. So that's generally going to be how a lot of these behaviors manifest in kind of these small systems, and actually mostly in social insects as well. A lot of these things, even if you think about consensus achievement, they tend to happen by kind of spatial distribution. So for example, I think it's cockroaches. They'll choose between two possible options by kind of aggregating in where seems like the best place. So it's not so much a conversation as it is a spatial distribution. So it's very important that we have a way to control the motion and the spatial distribution of our agents if we want to be able to build towards any of these behaviors. And so we started playing around with this. So kind of to start with, we were using, so for all of this, we've been using Volvox agents. So if you don't know what Volvox are, they're a type of algae. They are light responsive, which I'll kind of explore a little bit more in a second. But for these first two, so these first two, which are signaling and stigmergy, we kind of treated it more as almost like an augmented reality layer. So we're augmenting this behavior onto them. Obviously, they're not really aware of what we're doing, even though we are physically putting light onto them. So that is physically happening in the system. Obviously, we're not claiming that they are actually messaging between each other. But what we started playing around with is giving them these augmented light halos, so this projected light halo around the agent, which we could scale up or down the communication range of. So I've got the example here of 120 microns up to 360 microns. So I can play that now, and you can kind of see the difference then in how that propagates. So it should work that if an agent comes within that range, they then obviously start propagating with that range. And you can obviously see, as you'd expect, that for the smaller range, it takes quite a long time for things to propagate. Sometimes the signals would even die out as agents would move out of screen. Obviously for the 360, it's a bit of a wild spread. It's almost doesn't even really look like they're, it's almost just propagating through the whole collective because it's such a large range. Whereas for the, if I play it again, for the 240, it's a bit more of a controlled spread, and it looks a little bit more like the way that you might expect information to propagate through a group. So that kind of gave us a good indication of what might be a reasonable range for an agent of this size. So we then moved on to stigmergy. So this is kind of the pheromone deposition, which obviously is fairly self-explanatory. So we had these agents moving around and depositing these trails. What's interesting is that you might be able to see actually as the agents encounter the path of another agent, they do tend to slow down or even sort of slightly reorientate, but they all sort of do it slightly differently. So maybe I'll start that one again so you can have another look. So if you follow them as they kind of enter a trail, for some of them, they seem relatively unaffected, whereas for some of them, it really almost looks as though they almost get stuck in kind of a viscous liquid. So that was kind of the first clue. Obviously, we knew that Volvox did have this light response, but this was the first time really being able to observe it very well in our system. So that brought us on to trying to explore this motion control directly. So we kind of had these two building blocks where we said, okay, well, we can kind of in an augmented way have these signals propagate either through environmental deposition or through direct communication. Now, if we can start to really control the motion accurately, we can maybe start building towards some of these proper swarm behaviors like pattern formation, for example. And so the way that the light response is known to work in the Volvox is it is known to be an adaptive phototactic process, meaning that when Volvox enter a new, or they are illuminated with a new kind of light condition, they'll react very quickly, but then they will kind of, that will relax and they'll adapt to that. So that will become their new threshold value. So they obviously won't kind of continue adapting to that. So the way that we need to do it is probably through some sort of pulsed illumination. So some illumination time TI versus a relaxation time TR. But obviously trying to tune those parameters as finely as we could to get the maximum kind of stoppage. And with a system like this, it's unlikely you're ever going to be able to completely truly stop them in place, because they do like want to move and they do have this adaptive property. So we want to get that as efficient as we can to still allow for something akin to pattern formation or morphogenesis, even if it's in quite a flexible and organic way. And so we started by just giving this post illumination. So kind of just based on some crude just observation by eye, I tried to figure out what seemed like a good, and from reading the literature, what seemed like a good TI and TR, and we kind of went with that. So what you might notice, I'm illuminating only half of them, and that's obviously because I wanted to compare what happened with the illumination versus what happened without the illumination. And again, what's really useful about this kind of system is you can almost run those parallel experiments without having to start all over again and sort of recreate the exact conditions. They're all in the same medium, it's all done at the same time, but only half of them are undergoing those conditions. And so these were kind of the results, the velocity results that we found. So on the left is the one with post illumination, so those that are being illuminated, and obviously so the purple indicates a low velocity, the up to the yellow indicates a higher velocity, and you can hopefully see you do get this kind of periodic slowing throughout the population when you have this post illumination versus the ones that had none. But you can also probably see it's quite noisy. For some of them, they seem to be reacting relatively strongly, and probably you could even see that by eye as well, and some of them less so. So that kind of brought us on to thinking about what would be a good way to develop this further and actually figure out what is an optimum way to control these. So this is when we then brought on our master student Leah Matu, who did some work with reinforcement learning for us on this. To find a more adaptable control method. So obviously to implement this within the dome, it couldn't be super computationally expensive, and we also wanted it to keep it self-contained. So she explored a relatively simple tabular Q learning approach, rather than anything super complex and computationally expensive. We wanted to see what we could do in quite a limited resource environment. So obviously the idea to get the agent velocity to 0 through figuring out how to tune these parameters. So observing our system, tweaking the parameters, and seeing what seemed to be effective. And we want to do this on an individual basis. So treating each as an individual to ultimately try and affect a more collective outcome, since we noticed that they all seem to respond relatively differently. And what that looked like in practice, so you can see on the left here, we've got our two agents, agent A and B, and so they start off kind of in the same illumination pattern, both are illuminated, then both not, but then they unsync and they start to have these kind of different illumination cycles compared to the just crude motion control we did previously. And the initial results from this seemed promising. So you can see on the graph on the right, obviously, again, there's quite a lot of, it's quite sort of noisy, quite a lot of variance in general. But compared to either no illumination, continuous illumination, or just the blinking illumination we did previously, we were able to slow the Volvox down significantly. So you can see the velocity is a lot lower, and also crucially, the variance is a lot lower. So we found that by kind of giving them this individual treatment, we were actually able to affect an outcome where they were behaving a little more similarly to a collective. And so that's kind of where we got to on that so far. We're exploring that a little more now. So we now have another master student who's come on and is working on that and is going to try and extend that to figure out, based on this, how can we now start to implement those swarm behaviors I talked about before? So if we can stop effectively, and we can maybe do the signaling, for example, that puts us in a really good stead to try and do some pattern formation and some nice collective behavior. But thank you very much. Let me know if you have any questions.
Cool, thank you so much. Yes, maybe we have a time for one question. If there's none in the chat or raising hand, I have one. Can you try the opposite? I mean, instead of using the Q learning, blinking at them to make them stop, can you try to make them move around?
Yeah, so that is actually one of the other things that we did try was one thing we're kind of trying to explore as well is like the idea of herding them in a particular direction. So by kind of illuminating like aside because they're the way Volvox works is it's actually a whole colony. There's lots and lots of individual cells which kind of react in different ways. So actually, we've only done a little on that, but it does seem like we are able to, at the moment, we're trying to herd them up and then sort of down the screen. And yeah, it seems that is something that's possible. But again, it's fine-tuning the parameters, and they all, they're all quite difficult. They all seem to react in a very different way to whatever we do. But it certainly seems that that's a possibility. Yes.
Yeah, I mean, yeah, the noisiness of their response might mess up your Q-learning algorithm.
Yeah.
Okay. Anyway, thank you so much. Let's go on to our next speaker, Andrea Ferrigo, on the entanglement between evolvability and fitness in experimental study in Volvox, on voxel-based soft robotics, not Volvox. We just said that. Please.
Do you hear me?
We can hear you.
Okay. So now you should also see my screen.
Yes.
Okay. So I will now present our work on the entanglement between evolvability and fitness. So to start, what is evolvability? We know that evolution has created a lot of different species, yet much of this evolution depends on variation from a common ancestor, which leads to new species. For example, wolf evolved into domestic dogs as Pomerian, or my big black dog, Umbra. This property of a species to evolve into other good species is called evolvability. The fitness is a way more common concept, is the measurement of the capability of an individual to perform a task like catching the tennis ball. So unfortunately, we didn't work on dogs, but on soft robots, in particular voxel-based one, where the voxel are modular units linked together to form the robot body. The voxel can shrink or expand, respectively in red and green on the video, as a result of an external force or internal force. The external force is due to the expanding, shrinking of other voxel or the collision with the terrain, while the internal force is the controller that directly make the voxel expand or shrink. In this task, in this work, we use an open loop controller that you follow a sinusoidal function that depends only on the time, which is the one in the figure. And the two parameters, the frequencies and the phase, can vary among the voxel, and they are the parameters that we optimize. So the capability of the robot is measured in a locomotion task, where the aim is to move as fast as possible. The simulation is longer 60 seconds, and to make the task slightly more challenging, used a hilly terrain composed by the succession of slopes with different angles. We had two optimization settings, one where we evolve only the brain with so the controller with a fixed body, and one where we evolve both the body and the brain. We used two different evolutionary algorithms, simple evolution strategies, and quality diversity algorithm, which is MapElite, that I will now briefly present, as we will use it also for the calculation of the evolvability. So in MapElite, the archive that contains the solution discretizes a feature space, and there is a function called the descriptor that map a solution, an individual, into the discretized space. So, for example, if we have this grid bidimensional, so with two features of a size 3, and we have an individual with a fitness 1, we will insert, we have a descriptor which map the solution in a point, and if there are no other individual on the map for that position, the individual will insert. Instead, if the position is already occupied, only the best individual will be maintained for that cell. So if it is a maximization problem, here we have a robot with a fitness 2 that will replace the robot with a fitness 1. As I said, there is we need a descriptor for the solution. We use two descriptors, one for the brain-only optimization, which is the gate, that the measure is a measure of the frequencies on which the robot moves. In particular, lower value indicate higher frequencies, and higher frequencies indicate higher value indicates lower frequencies. In the brain optimization, we used a fixed body, which is the one in the figure here, and we divide the gate on the left, so the back, on the right, which is the front of the robot, as the robots move from left to right. In the case of the brain and body optimization, instead we use two features that describe the body of the robot, which are very simple. They are the width and the height of the robot. In this case, in the case of this robot, will be so 4 and 3. So how to calculate the evolvability? To calculate the evolvability, we maintain an archive similar to Mappelit, so with the same insertion policy as a Mappelit, but which is independent from the evolutionary algorithm used, meaning that the evolvability archive does not influence the evolution. At each generation, we try to insert the new offsprings onto the archive, and if in case we successfully insert, based on their fitness, we initialize their evolvability to 0. While the fitness of the parent, the evolvability of the parent is updated as shown in the figure, which is basically the average difference between the fitness of the offsprings and the fitness of the parent. So, for example, if we have this archive, we have this parent with a fitness of 1 that generates for mutation or crossover two individuals, G1 and G2, with fitness 3 and 2 respectively. We update the fitness, the evolvability of the parents to 1.5, as is 3 and minus 1 plus 2 minus 1 divided by 2. And we update, we add the offsprings to the map following their fitness, not their evolvability, as we will calculate later. Last but not least, the representation. So how we encoded the information in the genome and decoded to the robot. We had two representations, a direct representation and indirect representation. In the direct representation, the genome directly encodes the frequencies and the phase of the controller, in the case of the brain-only controller. While in the indirect representation, the genome encodes a set of parameters for two multivariate Gaussians that will generate the value for the frequencies phase for the controller. Similarly, we have used the same representation also for the brain and body, but in this case, the genome encodes also the information for the body. We see here in this example graphical representation that the bodies can be expressed as a grid of value, and each entry indicates that in that position the voxels are present or not. It follows in the same way in the indirect representation, where there is another Gaussian that generate the value for the body. Here there is another step that move from B to B1, which is the body, where we take the biggest contiguous area of voxel to avoid robots composed by two voxels that are not linked together. Okay. So we'll now see some results. First of all, an overview of the performance of the robot. In the left, we see the fitness trend, where in red, the direct representation, and in blue, the indirect one, with the dashed line that represent Mappelit and the solid line that represent evolution strategies. For the brain-only case, we see that all the methods reach similar performance, but the indirect representation converges faster. While in the brain and body case, the direct method produces faster robots respect to the indirect one. And as we can see on the video on the right, this is because the direct body are smaller and more irregular, and is more favorable to the locomotion. Here instead the results for the regarding the evaluability. Here we have 8 maps built with using the gate descriptor with the same size. In the case of the evolution strategies, so the first 2 columns, we recreated the map a posteriori using all the individuals generated during the evolution. In the upper, the first row, we have the result for the fitness, so the velocity over the x also. And while in the second row, the result for the evaluability. We can see that for the fitness, the robot move the limbs at higher frequencies, meaning that the best solution are in the lower part of the map. While concerning the evaluability, we see that is generally smaller than 0, indicating that individuals are replaced by individuals with a better fitness. We also see a good match between evaluability and fitness, where it means that where the fitness is higher, the evaluability is lower, because it's harder to generate better individuals. These are the results with the result for the evaluability with the body and brain case. We see, first of all, that here the maps are way more populated, but there are no area of high fitness, and this is because it's harder to make to optimize both the controller and the body. In this case, we also see that, in particular for evolution strategies, there are values that are positive. And this shows that these individuals generate better individuals in terms of fitness, but are not replaced by new and better individuals. And this can be an insight that we can take from this kind of plot analyzing the evaluability, because from looking only for the fitness, we see it can seem that also evolution strategies search all the feature space, while we see that in looking for the evaluability, that probably this area was not exploited, and only one or few individuals are generated for each entry of the map. This does not happen in Map Elite, where the selection process is random, and each individual in the archive have the same probability to be selected. So, in conclusion, we proposed the Map Elite as an independent support structure to analyze the evaluability. We tested it over 2 different evolutionary algorithms and 2 representations, the direct and indirect one, and 2 optimization settings, involving only the brain or the body and brain. We see how the evaluability is influenced by both the evolutionary algorithm and the representation, and we see that while negative values indicate offspring that are worse than the parent, the positive value indicate that yes, the offspring are better, but there is no replacement on the map. So the parents are not replaced. As a future work, we intend to extend this work in the case of the sensor evolution and learning. to see, and also to see how the vulnerability correlates with different features and tasks. Also, adding we want to add an historic analysis of the vulnerability, as now when we update the vulnerability map, we lost information about the vulnerability of the previous individual. We also want to incorporate the vulnerability into the evolutionary loop, in particular in the selection process. So, for example, selecting a set of solutions from the archive, the classic archive of the population, and a set from the vulnerability archive. Thanks for your attention.
Cool. Thank you so much. Do we have questions in the audience? Wait, maybe while you're writing. Have you tried other indirect encoding methods?
No, we didn't try other indirect encoding, even if we. So yes, we tried with a small multi-layer perceptron, but we don't make use testing because we see that the performance was very low.
Okay. I mean, the evolutionary adaptation performance, not necessarily the speed of the system.
No, if the robot generate using a multi-layer perceptron was very slow to me. They have a low fitness, and we have to look better to this process. But these are also other things that we want to consider.
Okay, cool.
As also a convolutional network for the body, for example.
Okay, yeah.
Defined as a grid.
I don't see anybody raising their hand. Somebody else has a question? I think you said it in the beginning. Oh yeah, there's one. How do you think the rugged landscape problem influenced your results?
A lot. Indeed, we want to check with other tasks what happens, as the illiterate to be used. Yes, is slightly more challenging than the flat terrain, but is still probably too easy for the robot. We mainly use this for to avoid the robot to vibrate, which is not a so plausible movement.
Okay. Cool. If we have no other questions, let's thank our speaker. Oh, no, we have one more. Since we have time, how do you divide the robots in bins and you map a lead?
Okay, so if we take, for example, these two robots, we define the space of the solution based on the picture. So, for example, the width and the height of the robot. We decide that there are 10 bins for each dimension, and then each dimension of the robot goes to another, as mapped to a position of the map. So, for example, if we have a map 10 by 10, each robot is 4 times 3, and we use the space in the body descriptor. We will put it in position 4, 3 on the map. I think that is clear. Okay.
Yeah. Okay. Cool. Then let's thank our speaker, and we go on with our next presentation from Marco Vigliani, pseudo attractors and random Boolean networks in single cell data.
Hi, the presenters will be
you changed
Gianluca D'Addese.
Okay. Yeah, I will
Sorry.
present it.
Okay.
Hi, Gianluca.
Perfect. Then anyways, please.
So you can see, no?
We can see your presentation. Yes.
The presentation, perfect. Okay, so hi, I'm Gianluca Dese, and today I will talk about our work, several factor in random Boolean network models and single cell data. Below, you can see our last article that talked more in deep about the concept and the result we are going to present today. For starting, we need to talk about what is a random Boolean network. That is the main model used in this article. As the name suggests, it's a network full of nodes, and those nodes are randomly connected, creating a direct graph. Furthermore, to every node is associated a state. It's a Boolean network, so the states are two, let's say on and off, or more simply 0 and 1. This model works in discrete time. That means that the state of the node at time t+1 is exclusively determined by the state of this input node at time t. All the updates of the node are done simultaneously. At the end, we have the truth table of every node that shows us the output of the node, giving the state of the input node. For the combination of input, there is only one possible output. Going forward, we have the dynamics of the random Boolean network. First of all, we have the attractor. After a first period of transition, the network tends to remain in one of these attractors. The reason the attractor are key focus in the study is the fact that each attractor in the genetic network is thought to be associated with a different cell type, muscle, neuron, or other tissue. The genetic network this way remain the same for all the cells, but the dynamic change depending on the tissue type. There is two types of attractor in the RBM. The fixed point that have all the nodes frozen in a single state, or we have the cycle attractor that have a certain number of precise states in which the network continues to loop. Then we have the dynamic regime of the network. We usually use distinct two main regimes that are order and the chaos. One of the differences, typically the order network tend to have fewer attractors than the chaos one, but the main point is that the order network tend to have a mild response to the perturbation instead of the chaos one that have a strong one. In the middle of these two regimes, we have the critical network. Those have particular relevance and subject of many studies because they have particular properties that have found to be also found in nature behavior. For the study, for this motivation, in this study, they are principal role in our experiment. That being said, the experiment in the RBN are not done evaluating only a single network, but typically an ensemble of them with the same properties to determine average behavior. In this work, we use 10,000 different networks for every combination of parameters that we can list here above. N is the number of nodes in the network. The experiment I will present have 1,000 nodes. K is the number of input of each node, and B is the bias applied to the truth table. Essentially, is the percentage of 1 in the output column. Now that I have explained the basics of the random network, we can go through the new concept presented in this article. We have a couple new ideas on how to see the attractor and the division of the network in two different parts. Starting with the average attractor, the idea is to compress the information of all the value of the node during the state of the attractor. It's easily computed by averaging the value of every node through each state. For a fixed point, the average attractor is equal to the basic attractor. But why we want to focus in so much on compressing this information? Mainly because one of the main limitations of the random Boolean network is to reproduce the real world is the synchronous behavior, which end up to creating many states and a lot of oscillation in the same time. And that doesn't really happen in reality. That's why we want to focus on the average node behavior for every particular attractor. For going to this to binarize again, we pass to the pseudo attractor. We will start from the average one and we'll define a threshold. Doing so, we can define what is under or equal the threshold that will be defined as 0, and what is above the threshold that will be defined as 1. Using now the concept of the pseudo-attractor, we can look at the network and all is a pseudo-attractor. Doing that, we can define what is the common C. That is defined by all the nodes that maintain the same value of state in all the pseudo-attractor. As you can see from the figure, the common C take most of the network, typically 70 or 80% of it. And the rest of the network is made up by the specific part, called island, just to match the idea of the common C. Those are nodes that appear to have different state in different pseudo-attractor. Having that in mind, we will now present our result of our work. Starting with the common C and the specific part, taking in observation the network with K2 and bias 0.5, that I can remind you that it's a network in the critical regime. We can see from the first graph that the size of the common part tend to be really high, typically above 60%. And on the right, on the other hand, we can see that how the size of the common part decrease with the number of attractors of the network. Doing so, the specific part tends to increase. Taking in consideration now only the critical network that is done by a precise matching of K and B, we can actually see that the criticality is not one of the points that determine how big is the common C. In fact, we can see in the first graph that the number of attractors decrease when we increase K, holding the criticality. And as before, in the right graph, this leads to an increase of the size of the common part. Going forward, we can give a look on our work on real data now. And we have taken an atlas of human cells with almost 600,000 cells and divided by 63 different tissue types and more than 27,000 genes. What we have made is taking all the single cell and of the same tissue and taking an average gene by gene of the raw count of this data. As I said, the idea is that every attractor corresponds to a type of cells. So the goal is to find and create an average profile to each tissue. So we can look at it and see if we can find or not the common C. To understand if it's this possibility to find it, we need first to discretize the real data. In the article, we use a couple of methods to do so, and now we present just one. Taking simply a threshold equal for every gene, we are going to binarize the value of the average tissue, typically using that threshold, using 0 if it's below or 1 if it's above or equal. This way, we can define what the common part is even in the real data. We can see increasing the threshold, the common part tends to increase. And the main concept here is to be able to create new constraint on the Boolean network model to reproduce the genomic network. As you can see, the real data, the common parts seem to be not really large as we see from the artificial network we see before. The idea so is to improve our model to try to match this type of behavior. That being said, it's necessary to expand our work on single cell data. One way we can do it is to expand the model to simulate a network with a number of nodes that go closer to the number of genes in the human atlas. So in this case, 27,000. After I've done that, we can focus on try different type of network, like scale-free or small world, and so on, and tries to see what match better this type of common part that we see in the real data using the discretization. And that's all. I was a little bit fast, but it's all.
Yes, thank you very much. Even you were a little bit fast. Do we have questions in the audience or in the chat? So I mean, in the meantime, I have one, because I think I missed that. Is the non-common part of your network non-common because it's sort of topologically not connected? Or is it, for lack of a word,
no,
logically not connected?
I can go back. No, not common is the fact that, as you see here below, is the node that have a different value in different pseudo-attractor. And the common part is just the fact that is the main part of it. It's typically 80% of the network, and the value of the node don't change the pseudo-attractor. So it's just a case that this is bright and draw like that they are separated.
Okay, so I mean, yeah, you're not, so to speak, getting inputs properly propagated into this for them to change with the state of the main one.
Exactly.
Okay, cool. Thank you so much. Do we have other questions?
I might have a quick question.
Please, Seth.
I'm not sure whether I understood, but so under N3 in that table, the number, let's imagine that in one of the pseudo attractors, it is a limit cycle. So you take the average value of that cell during the limit cycle, is that right? And so it might be 0.5, where it currently is at 1.
Mm-hmm. Yeah, but it's not possible to be 0.5 because this is a pseudo-attractor. So it's binarized by taking a threshold that becomes 0 and 1, taking threshold.
I see. So
because we start from the average that take exactly what you say, that
understood.
Everyone have a different number, and not for trying to compress.
So my question then is, sometimes if you go back to the first figure that you were just showing, with the common C and the table underneath, sometimes the number 1 in a column might not actually mean exactly the same behavior, right? It could be 2 limit cycles that both on average have a lot of on states, but they're not the same limit cycle, but they get recorded. Exactly. They get recorded as 1. So exactly. I guess that's pretty rare, but do you think that actually happens?
You mean that, wait, I cannot understand your question. Is like you have different state where different pseudo-attractor.
Let's imagine, we might not have time for this, but let's imagine that we have a cell and we put them in the common C, because in every attractor they have the value 1. But actually, in each attractor, they are going around a limit cycle, and we give them the value 1 for each of those limit cycles, but actually they are different limit cycles. So it's in the common C, but maybe it shouldn't be in the common C.
Because you say that sometimes it's go to 0. That's
yeah, that there are different patterns. Anyway, maybe let's pick this up on the Slack or something, because I think maybe it's a bit complicated to deal with.
I need to trust better this question. Yeah, sure.
Thank you.
And so it's a rather difficult question to sort of find out logical separability. Yeah. Okay. Yes, I'll refer you to the Slack channel. Thank you so much. And let's go on with our next speaker, Reiji Suzuki, a modeling and experimental framework for understanding evolutionary and ecological roles of acoustic behavior using a generative model.
Hi, can you hear me?
We can hear you, yeah.
Okay, thank you very much. Do you see the screen?
It's loading.
Okay.
I see it now, yes.
Okay, so okay.
I think you can start if you want.
Okay, thank you very much for introduction. I'm Reiji Suzuki from Nagoya University, and today I'd like to introduce a modeling and experimental framework for understanding evolutionary and ecological roles of acoustic behavior using a generative model. The emergence and evolution of acoustic interactions has been studied in artificial life, including the evolution of language. Also, there is an increasing interest in applying complex system approaches to ecoacoustics for conservation studies. Agent-based modeling has contributed to understanding the evolution of complex signals, and recent research focused on the evolution of audible signals. Here's an example of the audio spectrogram generated from the computational models, and this is a sound generated from the behavior of this small artificial creature. Such a emerging signals that is purely from the computational processes may have different types of complexity from those in natural ecological systems. That might make it difficult to discuss their roles in the ecological context, and it might also limit the interactions between the artificial and the natural systems. One interesting approach is to assume that direct interactions between artificial and natural systems, such as an installation based on the acoustic nature hypothesis like this. But today, I'd like to focus on the deep learning techniques that enables us to generate artificial objects that have equivalent complexity to natural objects. We want to bring this kind of complexity into the model. The purpose of this presentation is to propose a research framework for understanding the evolutionary and ecological roles of acoustic behavior by combining the modeling and the experimental approaches. This composed of two parts. First one is an evolutionary model in which the latent space of a generative model represents a genotype space. And then a generated object is regarded as a phenotype in the evolutionary model. And after conducting the evolutionary experiment, as a second part, we can further observe the role of the evolved phenotype in a real ecological context by conducting the field experiment. Today, I would like to introduce pilot and independent trials to discuss the feasibility of this approach, focusing on bird vocalizations. First one is the agent-based evolutionary model of syllables in zebrafinch songs. And the second one is a playback experiment on Japanese bush warbler in the wild. Let me note that these two trials is independent in the sense that we focus on different species. So the combining two approach is our future work. As a first part, we created a latent space of the zebrafinch syllable. This is a zebrafinch. This is a typical model individual, model species. And this is a song of this species. The syllable is a small component of each song. We created this two-dimensional latent space using a variational autoencoder using the spectrogram images of the syllables in songs. So as you see, there are cluster with the different colors. This means that the space well reflects the acoustic property of syllables. So each dot corresponds to each real sound in the dataset. And this also shows that the distribution of a generated sound, this also reflects the acoustic property of syllables. By using this latent space, we created an evolutionary model to see whether and how the complexity of spectral features can affect the evolution of syllables of zebrafinch songs. This is inspired by the model of a sympatric speciation by sexual selection. We assumed that the spectral feature of syllables could have effects on the mating preference of females. The model is very simple. We assume the male and female populations, each composed of 200 individuals. And each individual has two types of genotype. That is the two positions on the latent space like this, preference and syllable. As for the male individuals, this male expresses a spectrogram image as his syllable generated from using a syllable genotype. So this one and this one has different type of the syllable to sing. Then, as for the female, these individuals express a spectrum generated from a preference genotype as for preference for syllables. And this individual evaluates every male and selects a male stochastically proportional manner to the evaluation value. This is the equation of the evaluation value, and this component represents that the average difference in the pixel values between two images, so shareable and preference. So basically, this female chooses a more similar syllable to her preference. So in this case, this female might choose this one. And after this choosing process, this female produces a pair of male and female offspring. This is based on the standard crossover and mutational operations. This is the result. We assumed the 400 individual, and we conducted the experiment for 600 generation. And this is a distribution of the cerebral genotypes of males accumulated over 600 generations in a typical trial. Actually, the female genotype also shows a similar distribution. As you see, prezygotic isolation of individuals emerged through a kind of a runaway process in that the population segregated into a few subgroups. And also, in more detail, the population was basically composed of a majority group with a relatively simpler vocalization pattern like this. And also several other small subgroups that has more unique and complex acoustic features like this, or like this. So this is because the simpler phenotypes would be moderately preferred by many females, while complex phenotypes will be chosen by the limited number of females with high probability. So this is a very extreme result, but we can say that the complexity of spectral features can affect the evolution of syllables. This is a second part. In the playback experiment, we conducted a playback experiment to see if artificially created sounds using a generative model can provoke responses from wild songbirds in the forest. We focused on the Japanese bush warbler, this species, and this male of this species sings two types of songs, type H, this one, and also type L. So this song is very popular to Japanese people. And interesting thing is that type L song is a threat to rivals in the vicinity because territory owners frequently use this type in the periphery of their territory. So this individual tended to sing when some intruder comes into his territory. I will skip the detail, but we have created the two-dimensional latent space of songs using a variational autoencoders using some example recorded songs. And we created 25 artificially created songs from this feature vector. And then we conducted the playback experiment in open space in an experimental field at Nagoya University last year. This is an open space, and this is surrounded by the trees. And this is a territory of one individual. We replayed 25 artificially created songs on this individual in his territory. And then for each trial, we estimated the degree of aggressive responses against playback. We focus on two indices. One is the proportion of type L songs in the four vocalizations. In this case, that 0.33. And also we focus on the average change in the degree of arrival of this the songs at the microphone array, which is located close to the speaker. So the speaker replays the song of the artificially created sound. So it is like an artificial intruder. And in this case, that degree of change will be about the 80 degree. For these indices, higher value means that a more aggressive response from this individual. And then this is a distribution of the estimated aggressiveness indices in each case of the trial with different artificially created sound. This is a proportion of type L songs, and this is the average change in the degree of arrival of the sound source. And this is a directional histogram of the actual behavior of the individual in the experiment. So the blue one is a histogram of type L songs, and red is a type H song. So this figure shows that the individuals wandered around the speakers singing basically type L songs. Actually, that basically target male responded actively against artificially created birth vocalization. And interesting thing is that the song invoked the most aggressive response here is located close but a bit outside position from the clusters of the original type L song. This is a song. We think that its artificial but unique property of type L-like sound could bring about active responses. So the playback experiment with artificially created song can contribute to the better understanding of the role of spectral feature of songs. This is a conclusion, and the future work is clearly to combining two approaches. And we are conducting a pilot combined experiment on the spotted towhee. This is a latent space of the spotted towhee songs, and we conducted a similar evolutionary experiment and found the similar tendency of segregation in the evolutionary experiment. Then we conducted a field playback experiment on using this artificially created song, and the individual responded by this song, but the interesting thing is that he did not respond to the song at the center. So the average song may lack the essential property of their song. So sorry. We are going to do this kind of approach. Thank you very much for kind attention.
Cool. Thank you very much.
Thank you.
Do we have questions in the chat or in the audience? Yes, Seth. Oh no, Seth. Thumbs up, not raising his hand.
My version. On my version of Zoom, I don't have a button for raising my hand. So I do have a small question. So so this is great work. I want to find out a lot more about it. The example where you had quite a extreme bird song and it generated an extreme response. Do you think that that is similar to, I know there are studies of supernormal stimuli where you present an animal with something which is much more extreme than it would experience in their normal environment. So when you steal an egg from a gull's nest while it isn't looking, and you place a fake egg next to the nest and see whether it likes to bring it in. And if you give it an egg which is twice as large as a normal gull egg, they work really hard to try to get that egg into the nest, even though to us it clearly looks like it's not a real gull egg. So do you think that it was the relative extremeness of that that bird song, very different from the middle of your space, that made the extreme response?
Yeah. Actually, that's an interesting point, and actually we want to find out that kind of extreme case, and actually, yeah. So I think that this result, this is very preliminary, but this is interesting. I think that this song, this genetic song, might have some kind of extreme property. I think too extreme will be that. But if the sounds go too far, I think that it will lack the essential property of the this songs of this species. So I think that kind of extreme property might exist in between, this far too far and in the middle.
Yeah. Agreed.
Yeah. Actually, in this case, we also replaced this genetic song from around this around here. We could not any response from them. So we are clear about that this kind of a spectral property has an important information for this species.
Yeah.
But yeah.
Unless there's someone else who has a question, I also have a follow-up question.
I think you can go ahead, please.
Thank you, Aaron. In the first study, was there some pressure for the system to focus on simple calls? So I missed a little bit of your talk, but the result was that there were a lot of use of relatively simple calls, and there were some more complex calls. Is there pressure in the coevolution to use simple calls because maybe the way the distance algorithm, the distance calculation works, or why do you think simple calls were preferred?
Yeah, I think that basically the selection process is quite simple. The female compared with the spectral image between them. So maybe the distance from its own preference to the syllable. So I think that
Sorry, carry on.
Kind of normal songs can be preferred by the many females because not so much extreme.
Is that because they're in the middle? The simple calls are in the middle of the space, so they have on average the low distance to all the other calls.
Yeah, maybe not always in the middle, but in this case, the simple song syllables are around here, located around here, and we observed that this kind of major population tend to be located around here.
Interesting.
And
but some complex calls still survive. It didn't completely converge on simplicity, so that's kind of interesting.
Yes.
Anyway, thank you very much. Very nice talk.
Thank you very much.
Yeah, I don't have a question, just a comment exactly along this line of argument. Because you have sexual selection, of course, you get your runaway effect. And at the same time, because of sexual selection, you actually have a diminished diversity in the population. We did something similar like this, not with song, but with images, where one gender could create the image, the other gender could select the image. And they explore this image space quite well due to this runaway effect and at the same time the diminished diversity. So I would imagine that these islands actually swoop around in this space if you run this longer. But really my question more goes towards trying to replicate the experiment. If you run your evolutionary algorithm over again and again and again and again, I would expect different outcomes. Have you looked into sort of the variation of solutions that comes out of this?
Yeah, exactly. So this is an example, one trial, and if we do more, the similar but different distribution appears. But in many cases, that the majority population appears around here.
Oh, okay.
And once it from the initial population, the population evolve, and once divided into this kind of distribution, the population converges basically. So so the population does not so much wander around in the preliminary scenario, but it might depend on the settings of the mutation rate and or some other degree of selection pressures or something.
Correct.
Yeah, I think it will be very nice, interesting to see how this population wanders around, and that might be a future work.
Absolutely. Cool. Thank you so much. And let's thank all of our speakers. I think this concludes our session, and I think we continue at 2 a.m., right?
Exactly. Lunch for Europe, lunch break. See you later in 1 hour. Here again. Thank you, Arend.